Society of Critical Care Medicine, as well as X-Cures, and hopefully Mark will find his way down here for his presentation. But I think in the meantime, we can get started. I think, the success for CDRC is going to really be linked to how we engage patients and patient involvement and patient care. And in this case, generating data on a rare cancer, which is really the main proposal for the session today. And those diseases oftentimes, not just for rare cancers, but where there is what Vic had said earlier during this conference, those financial orphans, where there isn't financial incentives for many pharmaceutical companies to take these products and move them forward. And so the purpose of really this first session is to highlight a few of the key partners and partnerships that we have developed over the past year or so to identify the use of existing data to develop hypotheses that are generated based on clinical insights, as well as sort of any preclinical data that can help support that. And Bill will help shepherd that moving forward. In the second talk, that's where Denise will be talking a little bit about the role of registries and potentially, especially the patient impact on registries can make. And I think that's going to be a really key issue as we move forward is engaging the patient groups to see where we need to go. And then finally, the data technology component of this, extracting the relevant data that we need in a way that we can utilize it to be able to do the analysis that we want to do in the end. And then the last two talks for this morning session are really sort of more forward-looking. If we are going to be capturing real-world data to make some hypotheses and generate insights, at some point we want to try to confirm at some point through a clinical trial. And there are a couple of groups that have done this before in rare cancers and rare oncology. And so I've invited some of those to give some key insights of their challenges and what they've been doing and potential successes so that we can share those learnings as well. At that point, we'll break and then we'll come back and I'll kick it off with a few slides on what I think over the next year or so, what we would like to be able to do and sort of lay out those plans and get some feedback from you all at the end. But then we'll also have this panel discussion where we'll explore some of these subjects that not only we discussed earlier today, but also some other questions that you may have. So we want to make this, it's a small room and it's also an ability for us to really start asking some questions and having a conversation. So feel free as you go through it, write down your questions and be able to send that. And we'll discuss those. I don't think I have very much more to say other than I would like to thank you all for attending. And I'm going to ask Bill to come up and present his first set of slides. And we'll start the session a little bit early, but that will give us additional time for having additional questions. So just a green button, Bill. Great.
Is this?
Go ahead.
Keep going. Okay. Oh, there we go. So thank you very much. It's truly an honor to be here for me. We've been collaborating with the CDRC for about a year now, and it's something we're really excited about. So I'm here to talk a little bit about the why, which is sarcoma, which always brings in the question of how, and something that we always have to talk about in rare diseases, but what is the value proposition? And rare diseases are a place to innovate. We just often have to align a lot of people to do that. And that's something that we've been trying to do. Those are my disclosures. So I just want to bring everyone together to talk about what sarcoma is. So sarcoma is actually a heterogeneous group of cancers that arise from the bone and soft tissue of individuals of all ages. So it's actually the #2 solid tumor cancer in the pediatric population. It's rare. It's estimated to be about 16,000 cases diagnosed in the United States each year. I actually think that's a gross underestimation of really what's out there. But to give you an idea, those are similar in numbers to esophageal cancer, testicular cancer, and in those diseases, we have really well-defined treatment strategies. And in sarcoma, we don't really have that yet. And why? Well, sarcoma really represents about 100 different diseases, all which we now know have very unique biology and very unique clinical presentations. When we look at this list, we've now defined over 77 ultra-rare cancers that are included within sarcoma. So for us at academic centers, what we struggle with is how do we really begin to perform comprehensive drug development programs in these rare and heterogeneous groups of cancers? And I want to talk a little bit about the evolution of that, because we've really learned a dramatic amount through successes and failures over the last 10 years. This is a drug that I started to work with in about 2009 or 2010 now. It's olaratumab. It is a selective inhibitor to PDGFR alpha. Back in around 2010, we designed a randomized signal-finding phase 2 study of this drug with doxorubicin versus doxorubicin, our standard agent, in soft tissue sarcomas. And we just wanted to see that the drug could improve progression-free survival for about 2 months. If you look at the top curve, that's exactly what it did. It hit the hazard ratio as it was designed to. The bottom curve, overall survival, was really surprising to us. There was about a year improvement in overall survival versus our best agent, something we've never seen before in our disease. And surprisingly, and what was a wonderful outcome of this, olaratumab with doxorubicin gained accelerated FDA approval and became almost available worldwide. So it was a major advancement for us in the disease. Our community, which is a very tight community, got together very quickly and designed a really good confirmatory pivotal phase 3 study. And we brought with us all of the information from the trials we had just run in the last few years. And I think it was designed as well as it could be designed in sarcoma. So international, placebo control, there was a blinded component. We actually had dual primary endpoints looking at overall survival in all sarcomas, and then also in a leiomyosarcoma subtype. But if you look at the curves, it was a totally negative study. So it was negative in soft tissue sarcoma, in progression-free survival and overall survival, and it was negative in leiomyosarcoma in, again, progression-free survival and overall survival. But this was a major, major setback for us. Not only did we lose a drug that we believed had tremendous activity, but we also lost a commitment of well over a few $100 million into rare diseases to do research. So we regrouped. And we struggled. And we looked at this study as well as several other large studies that had been done in this time that were also negative in a similar patient population, over 2,000 patients with rare diseases. We tried to figure out why are these studies negative. And some of the biggest factors that came were the amount of variability that we actually introduced into these studies. So that study that I just showed you, the announced study, probably had 40 different subtypes of sarcoma enrolled into that study. That's probably 40 different diseases, right? So we're studying this hodgepodge of cancers in this. When you think about running a study like that, you have to run it in probably 15 countries worldwide, often 100 different sites. And when you think about the variability in referral patterns and practice patterns, that also comes into the study, not only when you have an overall survival endpoint, the available agents that are available for the various different disease subtypes after progression on a study like this, right? So all of that is going to come in and impact. It really highlighted too that we don't have the best outcomes in our disease and a best understanding a lot of these subtypes. But for me, what was really important is that we historically have not been comparing the same populations in really controlled early phase clinical studies where we see efficacy signals. And then when we move out through the regulatory process through phase 2 and phase 3, right, we end up studying different patient populations in these larger studies than we do early on. And we dilute the signals, we lose the signals, we lose opportunities for our patients. And I'm convinced we've lost a lot of good drugs because we haven't done this correctly. So we've adapted and we've really focused on ways where we can better understand the disease. And within the last 5 years, there have been all of these efforts to sequence as many sarcomas as we can. And this is just an effort of about 2,500 patients with various diseases. And what we see in these diseases now is tremendous genomic variability across the diseases, of course, but even within the diseases, right? So again, it's a little bit humbling because we have such variability in all of the diseases that we treat. But this has been important because when we began to see this and we took the knowledge from our prior studies that weren't positive, we started to say, how can we develop studies in very specific subtypes of sarcoma? And this was challenging because many of these subtypes, the truth is, are new disease entities that are recently genetically defined. We have world-class pathologists and they were forever calling us up and said, I found another translocation, this genetic event, it's a different disease. And then we as experts have to figure out how to treat it. But the truth is, in many of our diseases, we fully don't understand the clinical course, the natural history of the disease. So how can we develop a drug and how can we come up with endpoints that are meaningful for that patient, right? So we really need to understand how the disease affects the patient and what is meaningful for the patient. And I'll show some examples for that. Then we're dealing with a wide swath of drugs and technology that we have to understand their mechanisms of action. We then have to figure out how to do that in the context of the disease and the clinical behavior of the disease. So it's challenging, right? But it has allowed us to refine our approach to say, how can we do this correctly? So now we're trying to do early finding clinical trials where we can look at the efficacy signals and totally understand the patients that responded. And we do that in the context of a better understanding of the disease so that we can then develop clinical trial strategies that are, again, meaningful for the patient, the disease, and the drugs that were being studied with the hope that we can get more access to it. And I want to show 2 examples of this because it kind of highlights what often comes into this. This is a drug called pexidartinib, PLX3397, that I started working on again around 2010. And we designed one of the first basket studies. So after the phase 1 dose finding portion of the study, we tried to enroll patients who had diseases whose biology actually matched the mechanism of action of the drug, right? It was kind of a foreign concept at that time. So we ended up having about 6 extension cohorts in diseases. And you can see some nice responses in that, but nothing really dramatic. But there was one disease in there known as tenosynovial giant cell tumor that had these absolutely wonderful responses. These are the types of responses we dream of, waterfall plots and swim plots showing durable and sustained responses for patients. The problem was we didn't know a lot about tenosynovial giant cell tumor. So our community really began to scramble and say, well, what is this disease? How does it affect people? And how do we actually develop a drug for it? So tenosynovial giant cell tumor is not a cancer. It's actually a neoplasm where you have a neoplastic cell and it secretes colony stimulating factor 1. Colony stimulating factor 1 recruits inflammatory cells to a single joint, and patients get these large diffuse tumors. And it doesn't threaten their life, but it can totally change the trajectory of their life. It can deposit collagen, it can erode bone, it can bleed. So patients get decreased range of motion, swelling, and they really end up fairly disabled. This is a patient from UCLA that was actually a nurse that was on disability on narcotics, and she was about to have above the knee amputation. In the upper left-hand corner is a patient from Italy that was getting blood transfusions once a week because of the inflammatory nature of the disease. So we had a drug that could block this process, this CSF1 signaling. But this disease is very variable in every patient based on how it's affecting them and the joint that it's in. And not to mention that, but many people are dealing with this disease for 20 to 30 years. It's a disease that usually affects younger people in 30s to 40s. So we had to spend a lot of time with orthopedic surgeons who actually treat the disease, as well as patients, to understand how was the disease affecting them. And more importantly, if we had a drug for them, what kind of outcomes would be meaning for them? And we developed pain scores, and we tried to understand the natural history of the disease. And we actually developed a TGCT, patient-reported outcome tool, which was based on a PROMIS tool, which is a common tool that orthopedics use to actually determine how bone injury or joint injury is affecting the quality of life of patients, right? So again, understanding the patient's needs and partnering with the patients to do this. The other thing is that this is a really difficult disease to measure because it's a really diffuse disease. And if we use standard RECIST, which is a unidimensional measurements, we're often not even capturing the whole disease. So we spent a lot of time with musculoskeletal radiologists to develop a volumetric type of tool based on an MRI imaging that they used in rheumatoid arthritis to actually determine how well we were capturing the disease. So when you had that nurse who had a dramatic response after just a few weeks of therapy, you could actually capture the different responses that we could see radiographically. And importantly, because we had PROs, we knew in a non-malignant condition that those responses were meaningful for the patients. So we had some information, but then we were asked to design a phase 3 clinical trial from a small phase 1 data set. This was really challenging. We decided that we needed a placebo control, always controversial, but really important sometimes in rare diseases, because what it does is it can define the natural history of the disease, something that we don't know, right? So really important to that. But because we had a placebo, we didn't want patients to be on placebo for long periods of time. So we used a response rate at 6 months to determine efficacy. The problem is we had to use RECIST, which is, again, not a great response rate. And we were guessing statistically from a small patient cohort on a phase 1 clinical trial. So one of the things that we did to try to help with this is we added a lot of secondary endpoints, trying to look at patient-reported outcomes. We did goniometry measurements to see how the joint was moving. We looked at pain scores. And fortunately, though, this is a really strong drug for this disease. And we again saw these dramatic responses that were, 60, 70% over time. And these were durable. And when we looked at all the secondary endpoints, they were very meaningful for the patients. But the drug almost didn't get approved because there was a very rare cholestatic liver toxicity associated with it. And the study was designed in such a complicated fashion that there was a lot of missing data because patients had to basically do homework every night. Fortunately, it went to ODAC. The drug was approved, but this drug is still not available in Europe, right? So that's something that patients with TGCT in Europe don't have. And so some of the challenges, but it also highlights what we need to go through to understand rare diseases. There was a similar effort in another rare neoplastic process called desmoid tumors, desmoid type fibromatosis. And right around the same time, there was a trial looking at the drug sorafenib in this disease versus placebo again. This is also a disease that doesn't shrink when we treat it. It often scars down. So there was novel MRI imaging looking at it, as well as another PRO was developed known as the Goddess PRO. You can see in this trial, when you look at the waterfall plot up on top with the blue, you can see that this is a disease where the placebo was important because about 25% of patients had spontaneous regressions. If you didn't have that natural history of the disease, you would have overinterpreted the signal that you saw with sorafenib alone. But just like the TGC study, in which now there have been about 2 or 3 other drugs that are mimicking that design, there was also another trial in desmoid tumors that just came out positive with the gamma secretase inhibitor, using almost the exact same design, using the exact same imaging modalities, and again, using the Goddess PRO to actually show that the responses were meaningful in patients. So it can be done. It takes a lot of effort. Some of these were lucky where we had good pharmaceutical partners who could help us fund a lot of these efforts. But it's something that can be done. These, though, are neoplasms. They're not as genomically complex as many of the cancers we treat. And have we been successful? More so than we've ever been in my career. And this is just me thinking off the top of my head of drugs that have been FDA approved in sarcoma in the last few years, as well as NCCN compendium listed. And there's almost about 15 to 20 drugs on here. Never before have we seen that in sarcoma. So this has been a dramatic success, but it's bringing us to the next iteration that we need to have. As we're doing these subtype specific extension studies, what we're seeing in rare diseases, in diseases where we have no treatment options for patients, we're actually getting accelerated approval off of small data sets, which is really wonderful for us and our patients because we've had no treatment for them, but now we have something. But what it doesn't do is teach us the natural history of the disease. It doesn't teach us per se, how to use the drugs correctly for our patients. And I just wanted to highlight this disease, which is known as PEComa, which is a disease in which patients have a genetic abnormality somewhere along the mTOR signaling pathway. It's usually in a gene known as the tuberous sclerosing complex. And there are two genes, 1 and 2, that can be mutated. And patients develop these devastating tumors. This is a disease that we've actually used mTOR inhibitors, rapamycin, sirolimus, which have been around for many, many years, and we can see really nice results. This was with a new drug that was a NAB sirolimus, so it was a pegylated form of the drug that can last longer and is hopefully stronger. You can see here in about 30 patients, there were some very nice responses, a response rate of about 40%, and these responses were durable. And based on this, there was accelerated approval of the drug recently, which is wonderful because now we can prescribe it for patients. But what was interesting is some of the correlative work that was done on the study, where they looked at the genomics of all of the patients who came onto the study. And when you look at that spider plot down below, what it actually tells you is that the majority of the responses were seen specifically in patients with TSC2 mutations, right? So a subset of patients in a rare disease. And TSC2 is a very penetrant mutation in this disease. TSC1 tends to have a lot of other mutations that are also there helping the tumor, and it's probably why they don't respond. But now our community needs to understand the patient population, the genetic variations, the clinical presentations, and then how do we appropriately provide them the drug, right? So this is critical. The other thing is we still have rapamycin, and rapamycin is pennies to the dollar of this new drug. It's available worldwide. We don't know, does it have the same response pattern, a different response pattern? Does it have the same genetic penetrance and response patterns? So we have to begin to answer those questions, right? And that's really important. So we're beginning to see this shift in our community with this next iteration as we hope to get drugs approved on smaller data sets. We need to start early with some real good science and rationale. We need to be focused in our developmental strategy, looking to find homogeneous patient populations in rare diseases, which is really, really difficult. Very importantly, we have to partner with patients advocacy groups to understand how this disease affects them. What is the natural history of the disease? So when we apply drugs, we know how to do it correctly, but we need to take all of this information into the community so that we can actually design the right studies that we can run in an international fashion, not only to see early signals, but to validate those signals and not lose them over time. And this requires a lot of iterative work and a lot of correlative science, right? So this is now where our community is moving to. And how are we doing that? Well, we are a very collaborative community, and we are getting together across the world. And we actually formed what we call an ultra rare sarcoma working group, where we're now bringing together experts, multidisciplinary, pediatric and adult, to think about all of these diseases, to think about the appropriate design strategies and how to connect in the community. But it's a very slow process. We get together, we pick maybe one disease a year. We try to look in all of our academic databases to say, what information do we have on the patients? What are the needs? What drugs may work? We try to collect the natural history of the disease, and then we think about some prospective clinical trial designs. And we're moving the bar forward. But in the beginning, I said we have 77 ultra rare sarcomas. So I can't be 77 in another few years, be well into my hundreds and still doing this, right? We need to think how to accelerate the process. But the collaborative groups are together. The willpower is together. And we want to really change paradigms. And when we think about Europe, it's a lot different than the United States, because in Europe, if you have a rare disease, you usually end up at one of several referral centers within the country. And these centers are really collaborative. And that's where the patients get their care. It's very easy for them to pull data together and actually come up with an idea of how the disease is behaving. In the United States, it's totally different. Patients with rare disease often end up at an academic center. We see them early on, we confirm the diagnosis, and we often set a treatment paradigm for them. But then they go back home and they get treatment in the community. And then they may see another expert. They may go to a different center for a clinical trial. They may get a procedure at a different hospital because that's where the insurance allows for it. We also have patient advocacy groups that are bringing together registries. So how do we actually get all of these data together in the United States so we can have an accurate and complete history? And more importantly, how do we do it comprehensively in every patient with an ultra rare cancer, right? That's the most important thing that we can do. So we can begin to understand the natural history of these diseases, the clinical and genomic subsets of these diseases, as well as how do patients do on our best agents? What are our historical control? And within this, and why we're so excited to be partnering with the CDRC, is that there is tremendous opportunities to repurpose drugs. And it's interesting, as we were putting this together, we'll talk about repurposing some drugs like rapamycin, right, which we use in certain diseases. And even in our community alone, there's real controversy. Well, why do you do that? It doesn't work. Well, we use it all the time, right? There's no consensus in what happens. And if the experts are saying that, you can imagine what happens to patients. So we put together kind of a working paradigm of what we wanted to do within this collaboration. And the thought is, how can we get real world data comprehensively from EHRs in a patient with a rare disease from across the country, so we can understand the natural history of the disease. We can look for drugs that are responsive in those patients that providers are using all of the time. And then how can we actually disseminate that information, but then also think about using it for real world evidence? How can we use it to simulate clinical trials that will help us design the right clinical trials? And we put together this really wonderful working group with the CDRC. We have Memorial, which we're really committed to this. We've created a sarcoma working group of probably about 7 to 10 academic sites across the United States. We have patient advocacy groups, which you'll hear from today, and we're partnering with CURE, who is doing the data extraction for us. And conceptually, there's really 3 aims of this that I really want to talk about. The first aim is we want to establish a mechanism so that we can actually pull this complete data collection from multiple sources, right? We want to develop a comprehensive algorithm for extracting data on patients. And you'll hear about that later today, that it's totally possible, totally feasible. We need to be ahead of the curve with this, right? And when we do this, we have the expert groups together through the sarcoma working group and also the ultra rare sarcoma group with people who are interested in these diseases. And we can get that data back to them so they can begin to type out the natural history of these ultra rare cancers, right? The clinical and the genomic subsets. And importantly, we're coming up with a federated database, so this data will be open and accessible to people all over the world. This is not an academic venture where it's going to be curated at one institution alone. We're thinking of working with Eurocan, which is an institutional organization that can help us get the information out across the world, right? So it's a critical process, but we need to develop the infrastructure to do this and the algorithm to do this. So instead of doing 1 disease a year, you can have 10 working groups working on 10 diseases a year and just pulling that data in together. What's really important is when we start to do this, we can create the historical control data set, because in a lot of these rare tumors, we can't have randomized clinical trials. We have a lot of subsets where one subset of a disease may do better with the drug than another subset, just like I showed you in PEComa. So we need to see how patients are doing with our best agents, not only the natural history, chemotherapies, but also targeted therapies, like how are patients with PEComas doing with rapamycin. The other thing that you can do once you've actually brought this data together is you can actually begin to simulate clinical trials. The data is all there. You can change the variables, you can change the drug, you can change the presentation, right? You can change the variables, but you have the data, and that can begin to tell you how our patients do, just as if we were running a clinical trial. So we can begin to think about how to design the next iteration of clinical trials, especially when we have newer inhibitors coming from pharmaceutical companies that are more likely than not to show the signals that we want to see in the right patient populations. So in all of this is also creating drug-specific data sets. We want to combine those data sets to all of the tissue work that's being done that I showed you before. You know, again, at our institution alone, we have thousands and thousands of samples on patients that we've run genetic sequencing on. So how do we combine that and make that available? The final component is an important one. We need to do this in connection with regulators. And this is something that we're thinking about, because in my mind, it doesn't matter if we are coming up with clinical trials and we are coming up with outcomes that we think are important. If they're not things that are also going to be important for the regulatory process, we're not doing it correctly. So how do we eventually partner with this process with regulators, both FDA, EMA, regulators around the world, so that it's informing the process as we're going. This is not for specific advice, but to say, how do we begin to generate data that's going to be meaningful to help us shape the process in these rare diseases? And the other thing that we're working on is something that is, I think it's going to be controversial, but we're trying to create a master adaptive decentralized clinical trial that's available at almost any site in the world, in the US, academic sites and community sites, so that if a patient with a rare disease shows up in a provider's office and he's going to treat that patient with the drug that they have available to, we need to be able here in the US is one thing, but we can talk about, you know, what's available in London for patients with sarcoma and how many more lines of therapy I can provide them there. So how do we create, you know, the label changes, you know, the marketing approval where we can start to have these drugs for patients, especially ones that are helpful and cheap. So yeah, it's a great question.
Anybody else? Oh, yes, Sava.
Servo from Crowdfunded Cures. We're interested in the business model side of things. We're just wondering, like, is there any thoughts about like who would actually pay for these trials? Like, for instance, where you're doing a NAB sirolimus versus sirolimus, that would, if you could show the same efficacy, and NAB sirolimus is very expensive, you can actually have a trial where the trial has a negative cost for a payer that would do the trial. Have you thought about that sort of the business model side?
Totally. I mean, we unfortunately in this business, we have to think about the business model of things all the time, especially when we're going to pharmaceutical companies, asking them to invest in a rare cancer. I think to start, you know, it's not going to be that grandiose in the sense of let's do randomized studies. I think to start, these are going to be single arm studies with repurposed drugs. And that can range, right? It can range from things that are cheap like rapamycin, or in a lot of our diseases, checkpoint inhibitors or NCCN compendium listing, right? So there's also the cost of whether or not that drug can be provided to the patient. So that's the first part of the business model. The second part of the business model is, can we actually run these studies without a loss of cost for the sites that are running the trials, right? And I think there is a lot of funding and philanthropy available in specific diseases to run a specific arm. It becomes more complicated when we think about comparator to a potentially active control, just for the reasons that you said. I think a value proposition for a company, though, is that if we have a rare disease where we begin to subtype the natural history, understanding the genetic penetrance and the clinical variation, and then having enough data with real world evidence simulation where we think we have good tracks to develop a drug, it's a lot easier than for them to come in and invest in the platform for an inhibitor that they have interest in a rare disease, and they also get the international community. So it could be a way to gain that type of investment. But your question is good. The randomization is probably going to take time. What we could do, though, is have two separate arms in a drug where you're studying if people are able to use, you know, the pegylated one, and people are able to use the non-pegylated one. And then at the end of the day, you'll have some relative comparison, because both drugs are available for patients, right? And it may be that in certain countries in Europe and South America, they're using rapamycin, and the United States is using one. So, right, so it can give you that information, but it's still early on. And these are the things that we're beginning to think about. So I also presented this because throughout my career, you know, it's funny how many times people said, you can't do that. That won't happen. You'll never be able to do that. And 10 years later, we're a testimony to say, we have to do this, right? This is just the first iteration in the thought. And so these are why these discussions are going to be really important, to understand the barriers, the limitations, but where we can get started.
Hey, thank you. That was really interesting. I'm a pediatric oncologist, and I just wanted to hear if you could speak a little bit to the ways that you can approach the sort of operational challenges of kids being in, you know, even if they're over 12 and the regulators are, you know, happy to have them involved, that it's completely different sort of hospital systems, providers, et cetera. Have you guys thought about that?
I think we have to be able to extract data on, all of the pediatric diseases as well, right? So that's the first part, because in my mind, some of them are more organized in an understanding of the disease, but in a lot of times they're not. And I think we're still only learning the clinical variations. We struggle now with the clinical and genomic variations in rhabdomyosarcoma. What do they mean? What are going to be the prognostic factors in that disease? But in principle, it shouldn't be different if you're treating patients. Could you be treating patients prospectively on one of these small clinical trials and just collecting the data for us, right? And then how can we begin to include that in these larger data sets? So, at our institution, we've broken down the walls between adults and pediatrics, not only in how we practice, we're harmonizing all of our practice patterns, but we're also trying to break down the walls of clinical trials. So we need to get the age down. I also think that's important for the regulators. So when we think about that, though, and if we say, if all of a sudden, we're going to see an efficacy signal in 18 and above on a phase 1 expansion, but then let's move forward. And then there's a thought of bringing age down, maybe with drugs that we know a little bit about. What variables are we bringing into the trial that we should have some data to say, this is right, or, this probably should be treated as a different patient population. So it's really important that we start working early on with, our pediatric colleagues.
And that's where the real world data can come in.
Totally,
Really helpful,
totally.
really
Yeah,
helpful.
yeah.
So I think we need to, thanks, Bill. We need to move on, but I think that we can capture some of this and actually bring it back to the discussion later on. Yeah, perfect. Thank you. I think the next person is Denise Robinson from the EHE Foundation. Denise, thank you very much for coming. So Bill alluded to a little bit on the patient side on what we can do, and that's why we're hoping that you can help us on what you've been doing on the patient side and how we can learn from that.
Green button. Yep. Hi. Okay. Hi, everybody. I don't think that's my intro slide. Let's see. Oh, there we go. We'll see. I'm Denise Robinson from the EHE Foundation, and I have to thank Marco for inviting me to come here today, and Dr. Tapp for introducing me to Marco, really. And I would say that my goal is to be in Dr. Tapp's slides, in the earlier part of his slides, in all of those charts and graphs. And he says, and we learned a lot, and we did this right, and look at this great data. That's what we want to have in our disease. So thank you very much. So I want to talk to you a little bit today about the role of patient advocates in generating real world data. Our disease is very small. I think that we have a lot that we can bring to the table here to help clinicians, and specifically in the effort of drug repurposing. So the EHE Foundation is a US-based 501c3. The organization was formed by 5 women in 2015. It's a fairly small organization. We do serve an international community, though. We don't call ourselves an international group. We have international partners, but we serve patients now all over the world in over 80 countries. There are just over 2,500 people in an active online Facebook group. These people, they gen up questions about their disease. They tell us about treatments. We learn a lot from them. It's a support tool. What are we doing? So we are raising money. To date, we have awarded almost $2 million, which is not an insignificant amount of money for a little bitty rare cancer. We've awarded almost $2 million in grants and donations around the world for EHE specific research. We are initiating our own research as well as supporting collaborative research and giving those grants away. We're providing patient education and support. That isn't a core part of what our business model is on a day-to-day basis, but we have to do it. This is where people come, and we have to be there for people to help them. So that we're doing patient education for clinicians and researchers as well. There are plenty of clinicians out there who don't know anything about our disease. So that's an ongoing process. And then the least favorite part of the job is fundraising, but that keeps the lights on, it keeps us going, it keeps us putting money into research. So a little bit about epithelioid hemangioendothelioma. I actually became a part of the organization through a friend who was diagnosed, who was seen at Sloan Kettering. When I heard the words, I couldn't repeat them. I couldn't spell them. There are a lot of I's and O's and E's. It's very complex. Patients don't understand the disease either when they're diagnosed. And there are a lot of confusing definitions on the web if you start looking and you've gotten this diagnosis. It's an ultra rare sarcoma. Less than 1 in a million people are living with this disease. So you can imagine in countries not the United States, where people are literally on an island. They don't have care. They're all by themselves. It's marked by 2 gene fusions, predominantly the TAS-CAMPTA1 gene fusion, slightly more prevalent in women than men. Rare in children. I mean, we're already ultra rare, but we do have pediatric cases in our community. And recently we've seen just too many young adult women being diagnosed with this disease. And we have to uncover that. We have to get data to find out, is that something? We don't know. The incidence peaks at the 4th and 5th decade of life. So there's a burden of disease on these people, right? They may or may not be healthy enough to work and continue their life, but whether or not there's a physical burden of their disease, there's a psychological impact of their disease. As Dr. Tapp said, this disease is all over the place with its presentation. People can present with indolent disease with little to no symptoms. They can present with very aggressive disease early on. Liver, lung, and bone are the most common presentations of disease. And over 50% of people that we can tell and are reported in literature they present with metastatic disease. So someone comes in, maybe they have pain in their abdomen. There's a finding and you have this disease. You didn't have a lot of symptoms that would indicate that you would have gone in and had a cancer. There are no proven treatments for our disease. I describe it as buckshot if you look at what patients are reporting, but there are some retrospective studies that have been published. There are some case studies that have been published, but again, we need to gather that information. We need some systems to gather that information and prove what's working and what's not to keep going. As I mentioned, patients report significant psychological burden. There was an online study that reported that a couple of years ago. There have been no phase 2-3 randomized controlled trials. I would be surprised if we ever had those. There are 2 phase 2 studies that have published data that really didn't go any further. And then there are a couple of studies that were pending full data on those studies. So as we've talked about over the last couple of days, industry is really not that interested here. But also hospitals today, if we come or industry comes to hospitals with observational studies, there's a higher barrier of entry to get into hospitals to get a study done. So if any pharma wants to come to a hospital, we've been down that road. The clinician may be interested. The hospital says, thank you, but no, there's no benefit here. Our institution just can't run that study. Resources are very limited. As Dr. Tapp reported, there are 80 to 100 reported subtypes of sarcoma. So I don't think that patients think about this. I think about it in the context of my work that I do. Competition is a problem. We are competing for time. We are competing for clinicians' time. We're competing for expert care time at these centers. And it's really, really hard because if you think about it, there are a lot of disease groups out there, right? We all want to have experts help our disease group. And they're doing a lot of that on their free time. There is no free time. People don't sleep. But they're doing a lot of this on their free time. And then we want patients to get into those centers. We want patients to see specific doctors, right? So there's a lot of competition. There's competition for access to funding. And quite frankly, there are a lot of funding mechanisms for organizations that are our size. We have a lot going on in our foundation. I am also the only staff person in our foundation currently. So there's a lot to be done. So you need funding. You need to be able to access funding. So you're scrapping for grants. You're trying to find funding mechanisms, and then you're trying to network with really smart people who will say, yes, I'm interested in your disease. Let's study that. organizational capacity it's a limit. All of you who are involved in any advocacy at all, you know that that's a limit. We tell our patients, anyone who calls us, who gets this diagnosis, we say a lot of people are diagnosed in community care settings or in a country where they don't have a sarcoma reference center that's listed in Europe. And we say, please get to an expert center. You need to see someone who knows about this disease. But that is a challenge too. Barriers to care are very, very real. It may be that people don't have the financial means to get to those centers, but they also may not have the support mechanism to get to those centers. So it's a money and having that support. But then we also see, and this happens, we know our patients are going to those centers. They talk about a treatment plan. They may or may not get treatment at those centers. They may or may not enroll in a clinical trial that isn't specific for our disease, but it could be an immunotherapy trial. They enroll, but they do go back home. They get sicker and they ultimately die in their community. They're contacting us continuously as they're in those communities though, and they're saying, what else can I do? What can you tell me to tell my doctor to do? And those doctors and those patients are trying to call back to these expert centers and saying, help me. There's only so much that everyone can do in that situation, and it is unfortunate. And as advocates, I think all advocacy, but specifically in ultra rare advocacy, there's a large weight of all of this to get done. So, everyone says, stay focused on your mission. That's easy until 5 patients call that week and you are trying to help every single one. And then you realize that you haven't done the 10 other things that you needed to get done at the same time. But I think we're making good progress. The thing that I wanted to talk to you about today, and I'll touch on this lightly. So I think that patient advocacy is doing a good job in driving real world data and some of the initiatives that we're doing. So we started EHE 360. It was supposed to be an in-person conference. The pandemic had different ideas on that. So we did a virtual convening. It was the best thing ever. We did a virtual convening. Many more people came. So that was wonderful. That has continued as an annual event. And it's a collaborative event, including researchers, clinicians, and patients. It's a learning event. That's not data that's readily shareable right now, but it is an ongoing thing that is going to continue to help us drive data. The research grants and the giving away of money, investing in EHE research, I've talked about that a little bit. We started the EHE Biobank in 2020. This is a really big lift for a little bitty organization. And we have been successful. We have over 70 patients now consented on our protocol and have collected fluid, archived tissue, or fresh tissue specimens. And what we're doing, this wasn't the first thing we thought we were going to do when we started the biobank, but what we're doing is immediately getting those fresh tissues into a laboratory to see if we can get cell lines, at least some of it, and then archiving that tissue back in our bank so that it can be distributed for other research. We're not going to biobank tissue just for the sake of biobanking tissue. And now we're starting to decide what analysis we want to have done on those tissues. And that's all done at the Cleveland Clinic under the guidance of one of our expert advisors and pathologists. So we feel very good about the data that tool is generating for our disease. And then I want to talk to you a little bit about the Global Patient Registry and the Excelsior Outcome Study. So the registries, why I'm really here, I think. So we did a soft launch of our registry, and it's the first registry in EHE. We did a soft launch on Monday, and we have 10 patients enrolled, which is awesome, right? It's on the Nord IAMRare platform. And the benefit is that anybody in the world, anywhere, can consent and contribute their own data to this registry and tell their story. So if we can bring those patients that we know in Poland and Germany and France and, Chile, all those people together to tell their story, then we think that we can drive really meaningful, usable real world data that can be used in drug repurposing. That's the goal. I believe we're going to do it. So it's a natural history study. For those of you, and I think some of you are familiar with the NORD IMRARE platform, there was significant input from and discussion with the FDA and NIH on the development of the platform. It was one of the key reasons that we chose them as a partner for us. We did a lot of looking around at different industry and pseudo nonprofit providers for registries, but we wanted to have access to the data, and we wanted that data to be shareable in a meaningful way. So that's why we chose them. So in the registry, we're going to learn about, how this disease presents in people and how that disease changes over their lifetime. We're also going to talk about, we're going to have them describe in their own words what treatments they've had and what their responses have been to those treatments. We're going to use it as a way to identify doctors. This is a need. We learn organically from our patient population, but if we can ask people, where are they having interventions? Where are they getting treatments? Key treatments, that's going to help us. We keep a library of doctors that we know, that we feel comfortable with referring EHE patients to. So that's going to help us build that. Very importantly, we're going to identify people because we can describe them who might be appropriate for other research. So that's a key part of the registry. We want it to help better improve guidelines and develop recommendations for treatments. That's really necessary. Of course, it's going to continue to inform the EHE community, which I feel like is going to be so helpful. It'll get more people involved in the registry. It'll build trust in the registry, but importantly, it will answer their questions. It will empower them to know more so that when they go to their doctor and they can talk about things that they know and they have some data about that people are being treated. And then, of course, share data to support other research. That's why I'm here, really. Just to give some more credibility to what we've designed and what's going to happen in the registry, so you'll know what I feel like is the richness of information that's going to be included. So again, everyone is going to be self-reporting their EHE diagnosis. I realize this is going to be controversial in the clinical community, but we'll weed that out later. We need to start with gathering data. We're asking for people to self-attest that they've been given EHE as a diagnosis, not another disease, and join the registry. We're asking if they would like to provide evidence of their diagnosis, but that's not a requirement to join the registry. As I mentioned, it's on the IMRARE platform, and so NORD has defined 18 and up for legal adults. Those are the people who can consent themselves and join the study. Under 18 would be consented, and questionnaires are populated by a legally authorized representative. Deceased persons were very important for us to have included because we have so many people that come and pass through our community very rapidly, and we know nothing about those people. Dr. Tapp isn't going to have the data on those people either in his institution, possibly. So we need to gather that data. So those people can be consented and that data contributed by their designated representative, who would be someone who was involved directly in their care, who would know about their disease. So people will join at baseline. We're going to ask you to describe your diagnosis. Where did your disease present? What organs and body parts had tumors? Talk a little bit about how you came to your disease. Give your medical history, other cancers, other significant diseases that you've been diagnosed with. At baseline, we're also going to ask you about your current disease status. That's important because we have people in our community, amazingly, who were diagnosed and treated 20 years ago, and they're still alive, right? We have a lady who reported she had a liver transplant 20 years ago, and she's never taken immunosuppressants. And we want to learn about that. So these people are going to report their current status. There will be people who were diagnosed 2 weeks ago. So their diagnosis information about their diagnosis information is going to look pretty much exactly like their current status information, but we felt like it was important to get both of those time points. There are 2 validated pain assessments and then a symptoms questionnaire that we'll administer. And then treatment history, we jump right into that right away because that's one of the main goals of the registry, is to learn what treatments have been administered. More importantly, we want to know what patients thought about those treatments. How did those treatments control their symptoms in their own words? How did those treatments control their tumors or not, in their own words. And we worked with clinicians to really whittle down what those selections of answers are. And we feel confident that that data will be rich. So again, we're going to ask about hospitals and physicians at that treatment questionnaire. Again, this is every treatment you've ever had in your life at when you were coming in at baseline, and where did you have those treatments? And then semi-annually, so every 6 months, you're going to get a push, and you're going to be asked a lot of those same questions. You'll be asked again about your hospitals and physicians. You'll be asked if you're planning or considering surgery. And in our consent, that gives us a checkbox to recontact that patient to ask them if they might be willing to donate tissue from that surgery and talk a little bit more about that surgery. So that is an important part for us, and also to know where those interventions might be taking place. An important question we think is in the EHE status update. So it's not the most common question that's asked, but we see in our patient community, people who come to us, they have gotten an EHE diagnosis, they're in the community, they're dialoguing with other patients, and 3 months later, their diagnosis is changed to something else. People come to us by way of many diagnoses, but they also might leave us by way of a new diagnosis. And it's important for us to capture that. We ask on baseline if you had other diagnoses before you got to EHE, but then at 6 months, we want to find out if your EHE diagnosis has become something else too. That's important for us as we consider using this data and going forward. We're going to ask about new treatments, continued treatments, and any treatments you stopped and why you stopped those treatments. If you were on a combination systemic therapy and you stopped one of those treatments, we're going to ask you why you stopped that one treatment, and drop downs on all of that. So again, I feel like this is really rich data. NORD warned us. We have a lot of questions, but there's also branching logic in here. So there are a lot of questions, but it depends on where your journey has gone as to how long it takes you to fill out these surveys. Excuse me. So that's the patient reported side of things. And something that I've been working on for the last 4 years is trying to get patient data out of medical records, right? And so actually by way of this group, I came to be introduced to CURE's. We were on a call and CURE's was presenting their platform, and I said, that looks like something that could possibly help us. And we have not solved the issue of getting data out of medical records. And again, institutionalizing this, if I take this to 5 academic medical centers and say, I'd like to do a retrospective chart review study, that is wildly not popular anywhere. And we can't get it done. So we feel like this is going to be the way to get that done. So Excelsior is an outcomes registry. It's going to extract data from people's medical records. People will provide their consent. We will invite them to participate. So our foundations everywhere are a trusted resource to invite their patients to participate in research. So we'll invite people to join Excelsior. They'll provide their consent. They'll sign a HIPAA release that'll give consent for all of their medical records to be pulled at any institution that they've received care for their EHE. Really important because people are being seen at 2, 3, 4, 5 institutions, and we need to characterize that and create a history. So this is the way that we feel like that we're going to do it. It's a Q2 launch because all of this kind of culminated with the registry's ready. Okay, let's get that going. Let's get people in that, feel a bit of a break, catch a breath, and then we'll get the Excelsior study launched with those people who have joined the registry. The marriage of that data, obviously these are independent data sets, right? But we know who these people are. We can de-identify or code that data. And ideally, hopefully those data sets do inform one another over time. I want to mention one of the benefits of the Excelsior and the platform that CURE has is that patients can download their information. So if all of this information is gathered, right, and patients are maybe they're going to a referral center, maybe they want to seek new care, maybe they just want to put it in a binder in their kitchen. It doesn't really matter why they want it. We want them to be able to access that information. So we thought that was really important. And I also want to mention that researchers can access de-identified data. That was another key point for us. We don't want data that's locked up in silos. We need data that is shareable and usable. And we felt like this checked all the boxes. Dr. Tapp mentioned the EMA and sirolimus effort to get a label extension to have EHE on that label. That was just a recent kind of flurry of activity in Q1. And I just want to tell you by way of example. So, people ask, can you get your patient community to do these things, right? You have a registry, you're going to ask them to do that. You're going to ask them to participate in Excelsior. Obviously, we're going to ask them to, do 50 other things too, probably. But we recently launched a simple survey because we wanted to gain some feedback and get patients' perspectives on sirolimus for EHE. And so we basically said, okay, and over a weekend, we put together a little survey. We launched it for 13 days because we were rushing up into this application to the EMA, and we didn't have any data. We have no data. And we really wanted, not from the patient's perspective, Dr. Stacchiati, who was doing the application in Europe, has some data, but we wanted to have patients' perspectives and submit that with the application. So in 13 days, we had 122 people answer our survey around the world, which we thought was just great. May not, it may seem like a little bit, but we thought it was excellent. And people told us about how long they'd been on sirolimus, if they had stopped it, why they had stopped it, how well it had controlled their disease, their symptoms. We had 80 people who reported that they weren't on sirolimus. We opened the door slightly. We didn't say that you couldn't take the survey if you hadn't been on it or had ever been on it. We wanted to know your perspective if you were considering taking it. And that was important to us because 80 people did say that it was important to them that they have access to a drug if they or their clinician wanted them to be on it. There were people in our responses who said that their doctor wouldn't talk with them about it. These were mostly European people because they couldn't get access to the drug. So there's a lot of data behind this, but I just wanted to give this as a highlight of we have a very engaged community. We have people who are interested in telling their story. And I think now if we can capture that in structured data sets, that will be really important. We've talked a lot for the past 2 days. And when I made these slides, I didn't know who was going to be in the room, but a lot of you, maybe all of you are advocates. So, I'll just say in summary, we're a bridge, we're a gateway, we're a community. People need a place to come. And I look at the role that we play. We do have to support patients and families. We do have to drive critical research. I think in the context of this meeting, it is important that we are seen as a way to generate good quality real world data and contribute that data for other research. I want to hit on just a couple of things and I'll move along here. So getting, my priorities really in one, natural history data and the prospective outcomes data, getting those data sets populated so that we can get that data into the hands of researchers and clinicians and say, okay, let's study this further. Getting better outcome measures for our disease is really, or better endpoints for clinical trials. I think that's going to be, it's essential really for our success. Something that I'm really big on is persistent sharing. And I'm not talking about just sharing data. Sharing data is just one thing, but sharing lessons, sharing capacity, sharing capability. When you look at a lot of very small rare diseases, we don't all need, one FTE on everything. We don't have the ability for everyone to have a biobank. We do need our own data sets in many ways, but we do need to share and learn from each other. And I think that that's really critical for all of us to grow. My passion is that everyone should have access to genomic testing. It's not something that's widespread, but I think that as we continue to go down the road in research and we're sharing data and we're enrolling patients on clinical trials, it's going to be important that we do hand on heart know that these patients have this disease, that they have that gene fusion that has been detected, and it may or may not be sustainable for people to just be wandering around trying to find a pathologist. So that's my spiel on that. I just want to say thank you. If this is recorded, to people who contribute to research. We have funders. We benefited greatly from the capacity building grant from the Chan Zuckerberg Initiative. That was really a learning think tank. Aside from the money, it was a learning think tank. NORD as a partner and our partners globally. And thank you to the CDRC.
Thank you. Any immediate questions? So, one thing that you mentioned is the collaborative opportunities and the opportunities also to maybe coordinate better. And that's something definitely what CDRC would like to be able to do, is to not say, well, we don't all need to do the same thing, as you mentioned, so that we don't have one FTE at each institution doing the same thing. But then also to try to figure out how best to collaborate. And many of what you've said resonated well with what Bill was saying. So, knowing the fact that you guys are working together is one thing. And that's what we're really trying to do, is identify the different stakeholders in various different efforts and to coordinate them more towards what we need to get to. Because I think as we circle the room, we're all identifying with those needs, right?
Right. Yeah.
Okay. So, go ahead, David.
I love that you're capturing data on individuals who are no longer with us, because you can make a strong argument that those are the people we need to learn from the most, so that we can keep people with us longer. Designated representative, they don't have to designate that representative before passing. It's just someone within their, okay. So, and I think,
It's self-attesting.
okay, so they self-attest that they have a connection to that person.
That's
Could
right.
it even include a physician, or has to be a family member?
It is not required that it's a family member, but that's a good point. We hadn't thought about that.
Great if it could be
Yeah,
from clinicians.
That's a good point.
So,
Yeah.
Awesome stuff.
Good point.
Go ahead.
Thank you. Very informative. I have a question for you. As a patient advocacy group, how do you break through to find your champion doctor? We're obviously very small and we're representing clear cell sarcoma, but how do you find the institution, the doctor, the researcher that has interest in studying your specific EHE? How'd that come about? How'd your relationship start?
Bugging.
I'm good at that.
I mean, we are blessed to have a very active and engaged advisory board. And even beyond our advisory board, sorry, I'll step over here. Beyond our advisory board, I mean, there are many clinicians who are not on our board who contribute a lot of time. And I think that, when this is just my observation, clinicians see cases, they see things that don't make sense. And Dr. Tapp saw this. He saw my friend at Sloan Kettering. He didn't know it yet. We didn't know each other. You see things, they don't make sense, and you have to make sense of it. And I think that there becomes just a human nature to help. And that isn't inherent in everyone, but a lot of persistence.
Thank you. And thanks for the help you give to other advocacy groups. Too.
And if I can add to that, I think one of the most important things is to send patients to them, right? Because if people are in front of them with the disease, it's first and foremost on their mind. And it's this level of frustration of not being able to do anything. And then how do we move forward, right? And there's some great efforts that are going to start happening internationally in clear cell sarcoma. So tapping in to some of those and finding out who the investigators are. But send a patient, send another patient, talk to them saying, we're sending patients, what are you doing? And then really thinking about.
I send several to you guys.
Yeah.
Antonescu and I have a good relationship.
Yeah,
but
it's,
yeah.
and then we can talk, but it's really just building that up so that, the frustration comes in treating diseases that we don't have anything for from both sides, right? And so that's where the common ground is. So, yeah.
Okay, we'll talk because our patients are interested.
I think we'll go on to the next speaker, which is Mark Shapiro. So there's been several times that we've alluded to ex-cures, both from Bill's talk as well as from Denise's talk. So we'll hear directly from Mark. Thanks, Mark.
Great, thank you. I'm Mark Shapiro. I'm the principal investigator for the Excelsior Registry. I'm also the chief operating officer of a company called CURE. It was really nice to see when we started out kind of Bill's sort of overarching framework, and I think that helps explain kind of why I'm here. But CURE actually is a spin out of a nonprofit patient advocacy organization. And so I think a lot of the organizations here have cures in their name. We were really focused on how do we solve what we found to be one problem with that organization in terms of helping advise patients how to find the right doctor, how to approach kind of the next line of therapy for their disease. Oh, wow. Wasn't expecting that. So, we started with a mission, which is really to put the patients at the center of the research and data ecosystem, and really thinking like, how do we help them improve outcomes one patient at a time? And that vision really, it's involved in everything that we do, but our founder really wanted us to focus on creating a learning health system that put the patient at the center. Not something that's easy to do in a nonprofit. So we created a for-profit company and went out and raised about $20 million, which was not easy, in order to kind of address the technology barriers that we thought existed here, which was really that people came to the nonprofit looking for guidance on their care, but they really didn't understand their disease. And it's hard to give somebody ethical advice if they don't understand the disease. And that really took us down a pathway of trying to get at the source. So what that looks like today is a patient and physician facing web portal. It would technically be considered a personal health record, which is regulated by the FTC. But patients can sign up, they can consent, physicians can sign up and connect with patients. Patients can share their records with physicians, with members of their care team. And this is kind of an expanding thing. But what we set out to do was to collect and organize and structure all the patient's medical records. That turned out to be a very difficult thing. As some of the people in this room know, it was even harder than we thought it was going to be. But today we're connected to the health information exchange networks. There are 3 in the United States. They sit on top of dozens of health information exchanges, which exist at the state and regional level. And those exchanges exist to help institutions share records. Now, patients can access their records. So with the patient's consent, we can go collect those records. And then we've spent a lot of time trying to bring it into a common data model so that we could look at that continuum of care as people described. Patients are not, they move around, especially cancer patients. And as I'll show you a little bit, sarcoma patients, some of the other rare cancer patients that we've had some success working with advocates like fibrolamellar hepatocellular carcinoma. I think the average patient there had seen about 8 different cancer centers. So trying to put together kind of a longitudinal record, and their journey of even finding anybody who really understood their disease is hard. So we process that data. It's available back to the patient so they can log in, they can search, they can sort all those records, they can view the records, and they can share them. That data then is kind of a data catalog. So it's available for secondary purposes. One of the things that's part of our platform is consent to research. And so we can leverage that data both for the patient and for research purposes. For the patients, we use that data for trial matching. Like we're always looking and trying to help patients find clinical trials. I think for most of the patients that sign up with us, that's the ideal situation. It turns out to be super, super hard. I mean, I did clinical trials for 20 years. And once we started working with all these patients, we found that the main barriers tended to be things that were not on anybody's mind. Like we had a patient who was eligible for a pancreatic cancer trial, but her insurance wouldn't cover the out-of-network site. And that out-of-network site issue is a very common one. And unless you're independently wealthy, that will probably keep you from going to those sites, because this is a very expensive, any of these cancers is very expensive. But we try trial matching. We support clinical decision support. I'll show you a couple of slides. We work with a bunch of different organizations that are trying to develop algorithms based on genomics and clinical data to help patients identify, particularly off-label drugs that they might respond to. We do long-term follow-up and natural history study on that data. And then we can also do kind of an uplift on that data for, what we call regulatory grade or fit for purpose data. And we're doing some things right now. We're supporting some decentralized trials, expanded access programs, which is really what we started doing because we realized that trial matching was so hard. Was once you introduce the idea that maybe there's an investigational drug, if people screen fail for that, we went back to in 2018, originally one sponsor, now a bunch of sponsors, and we just asked them, say, hey, we helped this patient, they screened for your trial. Would you make this drug available? I think we've supported about 400 patients with compassionate use across the US. And we've had some really, there's some winners in there. Like, honestly, we've seen some really good stuff in there. And, unfortunately, a lot of things that don't go so well, but the data is there. And I'll show that because I think trying to turn expanded access or compassionate use data into something meaningful that could support an application is also part of what we want to do. So a little more about health information exchanges. This is really kind of amazing. It's almost like magic today. When a patient signs up, we can collect their records in 5 to 20 minutes. It wasn't always like that when we started this in 2019. It was taking us 10 days. So 5 to 20 minutes, we can structure that data and present it back to the patient in an organized way. I've often been asked, how do you know if it's all the data? And I think that's a really tough question to, to prove. About 90% of academic centers are connected. Most health systems these days, we get returns for more than 70% of cancer patients, and the average data return is about 1,400 pages of records. And I'll show sarcoma. Sarcoma is much higher. This is going up. We've seen a 10-fold increase in the number of connections in the last 12 months. The Medicare or MIPS program, there were a lot of carrots supporting organizations to purchase what are called certified healthcare IT. Now there's sticks, which means starting in 2023, there's a 9% reduction in Medicare payments if your organization is not connected to health information exchange. So we expect rapid uptake this year. Although the data that's being exchanged is in this old CDA format, which is, I guess, goes back to around the late 1980s. It's a lot of work to turn that into something sensible, but we are trying to turn everything that we find in there into FHIR data, including structuring genomics, pathology. We're doing natural language processing on path reports and clinic notes, really trying to find, for cancer patients, the important information, which is not that great in the structured data. And I'll show you some of that too. This is an example of what our portal looks like for a patient when you log in. So you can see we're getting hundreds and hundreds of documents, different hospitals, urgent care, telemedicine, labs. That's all searchable and viewable in the portal. But what we also found once we did this is nobody really has time to go through, 200 or 2,000 pages of records, even if you can search it. And that's why I'm gonna skip ahead of that. That's just showing that it only takes 5 minutes to get the data. We tried to present a visual cover sheet. So we settled on a Gantt chart, basically, of patient diagnosis, procedures, and treatments. So you just kind of see on one page what's going on, but you can still drill down into the details. And this was really a product of our initial work with tumor boards. So, tumor boards want records, but they really don't want to read them all. They actually just want somebody to give them a summary for their discussion. So we gave them access to the records, but we gave them a, kind of a high level cover sheet. And this, patients have access to it too. So maybe a picture is worth 1,000 words. I find this a lot easier to understand, conceptually. And then they can also generate timelines of treatments, timelines of diagnosis. We also see that the diagnosis changes. It becomes more specific over time, usually, although sometimes it completely changes. So Excelsior, we talked a little bit about earlier. Thank you, Denise. So we set out with a patient-centric outcomes registry. It's registered on clinicaltrials.gov. It's IRB approved. The goal was really to go out to patients and help them organize their own records and make that available for research. So today we work with about 60 different rare cancer foundations. I hadn't really thought that rare cancer would be the direction that we went when we started this, but through the efforts of some really energized patient advocates, we found that we were able to build significantly sized data sets. So the fibrolamellar hepatocellular carcinoma project started about, I guess, almost 18 months ago, through the efforts of one really motivated patient advocate, a gentleman who lost his son to the condition. And now we've wrangled a few other fibrolamellar oriented groups, and there are often a lot of advocacy groups. We do work with diffuse midline gliomas. There were 60 different advocacy groups, but they're finally coalescing into one now. It's a challenging thing because everybody wants to help, but it really is hard. There's a lot of emotion involved. But once you get that work going, now that fibrolamellar group, I think next year at ASCO GI will present more than 100 case reports aggregated. So kind of the whole longitudinal outcome. It's a very long disease. Those patients have the largest records of any that we've seen. But I think there's something interesting there. And then that data is also, one of the nonprofits has a fibrolamellar tumor board run by Dr. Paul Kent. And he, and that team actually, so we're capturing information on the rationales for their tumor board decisions, all going into the database. So we can kind of see, what do the patients get? How are the outcomes on those different things? So that's the goal with Excelsior. We do have raw source data that's available. So for academic research who want to collaborate with us, it's clinic notes, path reports. We're trying to scrape and structure all that stuff. We have the DICOM images, and I'll show you a project we're doing there as well. When we started, this was all facts. That was very hard. So we started with optical character recognition and natural language processing. Our two co-founders, computer science professors at Stanford. So this was an area of interest for them. So we still run natural language processing because generally the stuff we care about is actually not well structured in the medical record, but we do structure everything we can find. This is an example of when we were working with facts, what the system looked like. This is 2019 when we built this, and boy, natural language processing, we're in a totally different world today. What's possible now versus then is really different. But this thing has been trained by human abstractors going through more than 1 million records. It's actually pretty good at identifying different conditions, procedures, suggesting codes, identifying start and stop dates. We were looking for a common data model, and I come from the clinical trials area, and so we were looking at CDISC and CDASH, but because it's real world data, we ended up focusing more on kind of the M code structure, which the folks who developed that, super useful guideline for us. And although we now have a FHIR-based data model, there's a set of mappings from FHIR to M code. So if somebody wants M code, we can generate M code. We also use some of the OMOP tools from Odyssey, so another open source real world data initiative. We can produce Odyssey data as well for different users. We can also, because there are mappings between FHIR and CDISC, we could do CDISC as well. Our data also has a bunch of things that weren't really thought about in this. So we do quality of life surveys for patients, totally voluntary, but they're there. Some people like to do them. That includes, the EQ5D as well as the PRO CTCAE, which is an AE reporting instrument. And again, some people like to do that. And this data also has a lot of metadata associated. So, which institution was that procedure done at, and who is what's the NPI number? We have social determinant of health data as well, which can be useful because obviously that affects outcomes probably more than we really think about. DICOM images. And then we're also working to augment this through biomedical ontology. So when you see a term, those terms through the ontology imply other things, and we're augmenting that and noting those as augmentations to the data set. So glioblastoma is always a WHO grade 4, but if you don't see WHO grade 4 in the source data, right, we're augmenting that and noting that as an augmentation. That didn't come out quite right, but I think Bill showed this, right? These patients move, if they're lucky, they get, some early diagnosis or guidance from an academic center. Oftentimes what we see with people is, they're ending up late in their disease at academic centers. We don't know well, or we do now, but I think in general in clinical trials, we didn't know what was happening before that. And we have been asked actually by some pharmaceutical sponsors, could we go back and help them verify like how many lines of prior therapy? Actually, it was it was during a FDA submission, the FDA asked them, how many lines of prior therapy and did you verify that? And they said, well, we didn't verify it, but that's what the patient reported when they came in. So we went back to see if we could help them verify that data. We've been doing some research on our data. But I think our point here in doing the research is really we want to show that you can do research. We're not, I have 2 data scientists on my team. We make this data available for research collaboration, and that's what we really want to encourage. But we want to show people that it can be done. This was an abstract from SNO last year. So this is the diffuse midline glioma cohort, about 150 patients in total. It's rare pediatric brain tumor, about 200 cases per year. And a simple multivariate Cox regression model. And what we see here, which I think is relevant to repurposing, is that in this model, right, we're seeing what we expect, which is like that the anatomy of the tumor location matters, right? That the prognosis for Pons location is notably worse than the thalamic node location, and that the pediatric patients have a worse prognosis than the adults. But also what we saw in this, there are about 60 patients who were exposed to an investigational agent through the sponsor's compassionate use program. And you see here, a significant difference on overall survival that holds up under this multivariate analysis. So, even taking into account some of these other factors that affect the prognosis. The other thing I would say about that cohort that's interesting to us is a lot of those patients screened for the trial and screen failed. They were, too sick, too low performance status, and yet we still see an effect there, which, for us was heartening, right? This is a promising agent. Another project we're doing with the University of Wisconsin. So we collect the DICOM images. Most patients have between 5 and 15. In this project, we were just trying to isolate the tumors and separate out edema for glioblastoma patients, and then quantify mass effect, which is how the brain is deformed by the presence of the tumor. And my point here is really that even though this is real world data coming from an EHR, that if somebody wanted to take that data back for PFS, right, you could do an independent radiology review. We can de-identify this and send it to a central reader. This is an example of something that we're doing with the Nonprofit Institute for Systems Biology. And here we're sharing this data for patients who want this service. It's a pathway-based analysis of the tumor molecular sequencing. And they use it they have predictive algorithms based, I think, on data and cell lines and labs. The point here, which was kind of interesting, and this is another SNO poster, but for this particular patient, they predicted a response to lomustine, which is not a widely used treatment for glioblastoma anymore, although it is approved for it. It's mostly been replaced by temozolomide, but they predicted a response in this patient. Patient had over 2 years of disease stability, which is a markedly good outcome for that treatment. Conversely, after they progressed, they didn't actually see anything. They predicted a few things, would not have an effect, which is what they ended up trying, and that patient wasn't on for very long. But another example here is the swimmer plots for osimertinib, where we just looked back retrospectively with them in our data, ran this analysis, and could see that, again, this is an off-label, so, potential drug repurposing scenario. The patients who were predicted to have a response had a, considerably longer time on treatment than the patients who were not. And we're actually working with them now on a prospective study. This is a replication of a randomized clinical trial that we did. This, I'm proud to say, took 2 days. So the data was already structured, which is great. We were able to generally match the cohort in the clinical trial, which was completed. I think the 5-year survival was published in 2015. Our patient data was coming from 2016 and afterwards, so totally separate cohort. In the real world, remarkably similar overall survival results, which, for us was encouraging. I think actually for the manufacturer of that product also encouraging, because they have not done any research since they got their approval. But I can say that, we've seen some interesting data here, and I think it's, it's encouraging that the survival benefit holds up outside in the real world. So we're excited to be working now with C-Path on this initiative. I think our role here is to try and help make data available for, the various stakeholders. That includes our new partnership with the HE, which we're very excited about and getting started now. Congratulations on getting your registry going. And the initial focus is going to be EHE and PE coma. So I did a little bit of a quick look last week on kind of what data we had, because we hadn't really been thinking too much about sarcomas before. So this is a snapshot of how many patients we have so far in our database and where they are. You can see here too that the patient encounters are concentrated more around cancer centers, but the patients themselves, especially in the Mountain West, have to travel a lot farther for care. So this was a big surprise to me. 400 people with a coded diagnosis of any type of sarcoma, scattered all over the US. A lot of records coming back, that almost 3 times the average cancer patient. So we haven't really done anything with this, but we're excited to know that there's some data there. The coding data or coding of sarcoma data in EHRs is not good. So, take all this with a grain of salt, but these are all coded diagnoses, and the counts of the number of patients with those diagnoses. There were angiosarcoma, there were 1 or 2 EHE patients in here too. And they're also, just to show you what could be done, looking through all the coded diagnoses for sarcoma and what the medications that were being given, we're seeing interesting things. So there is some pembrolizumab use and some of the other agents. I hadn't actually thought about it, but seeing Bill's presentation too, we have an interesting cohort of chordoma patients on tazemetostat, and are going to try and publish that next year. Most of these patients actually are getting genomic sequencing, which we agree is very important. If you have a rare cancer, almost certainly need to get this done. Almost half of them had somatic testing, and about a third had germline testing. But we're able to get all of the data from these various genomic labs, which has really opened up in the last year. And just, we tried to do a quick little coding. You can see most of this is leiomyosarcoma, but there are some more rare sarcomas represented here, including epithelioid and synovial sarcomas. Again, I would want to go through and parse the path reports and stuff before I said anything definitive, but we know that there's some interesting information here, and we're looking for partners in this room, hopefully, to help get more data and analyze this data. That's my last slide. So thank you very much.
Thanks very much. So are there any quick questions? And I actually, just before you go, can you go back to, I think, slide 74? I noticed something interesting. Yeah, so that's why I got up. In anastrozole.
A lot of gynecologic sarcomas. I mean, that was definitely
That was primarily because of
and
that.
so that, I mean, that's my guess, is that they're treating it like uterine cancer,
And it's just,
and that's kind of what they start before they realize what it really is.
so there's going to be quite a bit of data on that then, I guess. Well, 22, the most that we have.
Yeah, and again, we haven't really rolled this up by, so this dashboard's only a few weeks old. We're now rolling it up by active ingredient rather than the actual code. So you can see there's different codes for different regimens of anastrozole there, or brand versus generic.
Generic, right.
But the data's in there, and I think for us, if people have interesting questions, we're excited to answer them if we can. We've got a real interesting poster at ASCO, which in that one, I mean, we found an over-the-counter product that was interfering with chemotherapy in our glioblastoma patients. It was a dramatic effect that held up under all the subgroup analyses that we did. So we know that the data is in there, and that there can be meaningful stuff done with it. Any other questions?
Maybe in the interest of time, then we'll go on to the sort of the next couple of sessions, which really start taking a look at the clinical parts of this. So if we capture the real world data, what are some of the bigger challenges of actually going into the more clinical pieces of this? And there are 2 speakers that I thought about as we were coming around and having this, putting this together. One of them was Claire, and the other one is Vigila. So I'll have Claire come up and talk about some of her experiences with the challenges and opportunities.
Great, thanks, Marco. And I'm going to apologize in advance if I go through this really quickly, because I do have to leave right after to catch a flight. So I'm probably a little bit faster than I would normally do, but I did want to talk about some opportunities that we've seen and we've participated in to catalyze impact in rare cancer trials. So I really loved on Monday or Tuesday rather, how we all started with our definitions, because this is a slide that Cures Within Reach puts in all of our all of our presentations about what we consider to be repurposing. And I know that the focus of this conference so far has been drug repurposing, but Cures Within Reach includes drugs, devices, nutraceuticals, diagnostics. So any therapy that's been approved by a regulatory body, whether it's the EMA, the FDA, Health Canada, what have you, taking those and then testing them in a new indication. Different, of course, from repositioning or rescue, which can be a human safe compound that has not yet had a regulatory approval. And we are open to both philanthropic repurposing opportunities where there might be a great opportunity for patient impact, but not as much opportunity to make a profit. And also commercial opportunities, which could be taken all the way through to regulatory approval. Again, so the mission of Cures Within Reach is to leverage the safety, speed, and cost effectiveness of testing already approved therapies in order to improve patient quality and length of life. We've been doing this for about 15 years now. We are disease agnostic, geography agnostic, institution agnostic. What we do is we work with our funding partners in their particular areas of interest, strategic interest, and then we'll issue a call for proposals for investigator-initiated studies and make selections from there. And there's a lot going on in this slide, but really the most important thing to know is that the types of trials that we really like to fund are these proof of concept, phase 1, maybe phase 2A trials that just need some data, need to answer a question so that they can go on to a bigger study. This is what we call the come back when funding, when a PI goes to the NIH or American Cancer Society or DOD or what have you, and they say, we've got this really great idea from real world data, from preclinical data, from case studies. We'd like to do a clinical trial. And they say, come back when you have some pilot data. And that's where we come in. So we find that this is a really critical stage to fund. We're not funding all the way to the end. These are not huge dollar amount grants, but they can play a really critical role. And again, we are indifferent to the commercial value of the projects that we fund. What we focus on is patient impact. That's our key driver in what we do. So, we do have disease agnostic portfolio, so you can kind of see the different areas that we do have ongoing trials in right now. We have currently 33 or 38 ongoing trials in 27 diseases across 7 countries. So we do fund internationally. Rare disease is always at the top of our portfolio, and that's consistently 45 to 55% of what we do. But also right up there is oncology. It's usually 20 to 25% of our portfolio. And there is overlap between those two categories as well. So we've done a lot of work in oncology over the years, and I just want to share a couple examples of some success stories we've had. And the first one is actually one of the very first repurposing studies that we funded, and it actually is what made us focus our mission exclusively on repurposing research, and that was funding Dr. Raj Kumar at the Mayo Clinic in his early studies looking at thalidomide and multiple myeloma. And there's a great example of how, we didn't fund all the follow-on work and the regulatory things that needed to get done, but it was a label change. It was FDA approved and has become a standard of care for these patients. We also have 2 device examples that I can share. Again, not just drugs for Cures Within Reach. One is looking at a laser device that's used in liver cysts. This was in prostate cancer at the University of Chicago. They were then able to raise over $2 million from the NIH for follow-on trials. Similarly, there was a ablation device at Virginia Tech in conjunction with the University of Louisville. They developed an online training tool that's available to clinicians and researchers about how to use this device and optimize it for pancreatic cancer specifically. In addition, NIH gave them the grant to expand their work to include liver cancer as well. And it's not just governmental funders, it's also industry. There's another trial where we were a partial funder. We weren't the full funder for this trial, in combining two immunotherapies that are approved in melanoma for lung cancer. That gained a lot of interest from industry, and they are now sponsoring the follow-on trial. So you can see this catalytic impact really does have an effect. And I can tell you that over the years, we've invested $8 million in various repurposing research projects. And our funders have gone on to raise over 80 million total in follow-on funding. So now I'm just going to share a few examples of what we hope to be future success stories in rare cancers and how they were trying to increase impact in these areas. The first, since it's a sarcoma group, I'll talk about our osteosarcoma program project. This was actually an ongoing trial already when it came to Cures Within Reach, and it was the co-winner of our Cure Accelerator Live pitch event. So for those of you who don't know what Cure Accelerator Live is, it's a really fun event. Think Shark Tank, but instead of entrepreneurs, you have clinical repurposing trials. And we feel like this is a really fun way to bring this type of trial to the public. Everybody gets to vote on the winning project, and it's very interactive. We've done this live and virtually. But this ongoing trial is looking at hydroxychloroquine in osteosarcoma. And the PI that we're funding had this idea to take the samples and look for biomarkers, but they needed a little extra funding. And this is where Cures Within Reach stepped in. So this will allow the team to look at the samples and see if there are biomarkers that can not only tell why some people respond to the trial, but why others don't. So again, kind of getting that little bit of extra money to add another study to make this data more robust and applicable to clinical care. Another example also happens to be at MD Anderson. This is a newly funded trial for us. This PI was already running an adult trial in AML, looking at metal reduction therapy for these patients, adding it to chemotherapy. And based on the initial results from the adult trial, plus based on some data in the literature, she really wanted to expand it to pediatric, young adults, and adolescent patients. So again, this is an opportunity where Cures Within Reach was able to come in with additional funding and allow her to expand that. So again, now we're increasing the impact of a trial that's already funded by somebody else, but now we're going to have a wider range age group for these data. And the really great thing is that her IND is already approved for 18 and up. The young adult category is, that 18 to 25 range. So she can immediately begin focused enrollment in that age group while she works to include pediatric and young adult and adolescent patients as well. We also have seen that our funding, you don't have to wait till you're done with your study before a PI might get interest for additional funding or additional support. This is a trial that we funded out of the Children's Hospital of Philadelphia in pediatric ALL. When we originally funded it, it was single site, 10 patients. Again, we're not funding huge trials, just pilot data. And after she was awarded funding from us, she started to get interest from other colleagues in the field. It is now expanded to a 7-site trial that's going to enroll 40 patients and has attracted support from industry for study drug. So that's a really great problem to have. And I kind of talked about this a little bit in the panel yesterday. The downside, of course, is that now we're dealing with 7 different IRBs and trying to get everything coordinated. So it has extended the amount of startup time, but the increased patient impact will just be, we think, really important. And again, going from 10 patients to 40 patients is really, really huge. And it's not just these types of trials where we go out and look specifically for rare cancers or something like that. Cures Within Reach also has an initiative in diversity, equity, and inclusion. And in this case, what we're looking to do is in the US, increase the diversity of clinical research, not only in terms of the patients that are enrolled, but also the PIs who are conducting the research. And so we had this project come to us from our DEI initiative, and it's looking at squamous cell esophageal cancer, which is more common in Black patients, and Black patients have poor outcomes. And this is another device project where this PI wants to take a device that's normally used to screen for adenocarcinoma, which is more common in white patients, and make a routine screening protocol for squamous cell carcinoma. At the same time, tying it to some social determinants of health to identify high-risk patients and make sure that they're involved in the screening process. And what's a little bit different about these health disparities grants that Cures Within Reach has is that we are also adding supplemental funding so that the PIs can work with community-based organizations. And we talked a lot about patient organizations, which are really important, but, and I think we heard this from Cynthia on the panels yesterday, talking about long COVID and patients of color, that it's important to work with organizations that are known and trusted in these communities to help support enrollment, to help support engagement, to help build trust, and to also share the results of the trial once it's done. So we are providing funding for those activities that are separate from the clinical research activities. And they can be community-based health organizations, they can be faith-based organizations, they can be social services. We have a project that is not yet started, but has been approved, looking at breast cancer with Black women. And that PI plans to work with beauty salons. It's wherever the patients are and what organizations are important to them. Those are the relationships we're interested in cultivating. And then finally, we've talked about this a little bit, that cancer is not a strict US or North America or European problem. It's a global issue. And cancer burden is, of course, higher in low and lower middle income countries, or LMICs. So again, Cures Within Reach has a specific initiative that we call ReGrow. It stands for Repurposing Grants for the Rest of the World. This is a funding opportunity that's specifically for PIs who are based in LMICs. The idea being that, if we can grow clinical research capacity while at the same time finding generic, inexpensive treatments that are accessible and available to patients in these countries, that's a good thing for global health altogether, and a great example of how repurposing can really make a difference. These are disease agnostic calls for proposals, but I can tell you that, everybody thinks, oh, you must get a lot of infectious diseases or neglected diseases submissions, and we do, but right up there with them is oncology. It's either the top or the second most popular one that we get in any given call for proposals. So this is a project in triple negative breast cancer that we're supporting in Nigeria. In this case, it's adding clofazimine to the standard of care in order to help improve outcomes. And, I think the real lesson or learning that we had that I'd like to share with the group, and I hope you all take away, is that this is a really great opportunity to engage stakeholders that are not here, those that work in low and lower middle incomes countries specifically. We had no connection to LMICs when we started this, but we knew people at the Fogarty International Center at the NIH. We knew people at Global Health Catalyst that works with cancer specifically in low and lower middle income countries. And we worked with those organizations to help us build relationships with institutions in LMICs to help find the projects, because we knew if we went knocking on the University of Nigeria's door, that they were going to be like, we don't know who you are. Why should we trust you? Why should we apply to you? But when a trusted partner again says, hey, you really should check out what Cures Within Reach is doing, we found that method to be very successful. So again, I know this is really fast and I apologize, but just again, to summarize these opportunities, there's a lot of opportunity for collaborations between researchers, and also we just want to encourage researchers who already have a funding source, if they need additional funding, Cures Within Reach is open to that. So are other funding organizations. So think about where you can pool your resources, even if you already have an ongoing study. Think about patient engagement, certainly with patient advocacy organizations, but also beyond that, and look for health disparity opportunities in rare cancers. Again, pediatric sarcoma is a great example of that, where it's more common in minority patients. And then finally, think about stakeholders globally that you can work with in order to address rare cancers. And maybe it's not always funding, maybe it's brain power, maybe it's connections, maybe it's really interesting research in LMICs, because they have no choice but to repurpose, because they don't always have access to the latest drugs and the most recent therapies. So broadening the scope of how repurposing can help cancer patients globally. And like I said, I do have to run in just a minute. I won't be able to be here for the panel, but please reach out to me, come visit our website. I'd be happy to talk to anybody else more specifically. Thank you.
Thank you very much, Claire. Are there any burning questions, or can we move on to the next speaker? Because we're running a little bit late, so maybe what we can do is get Vigila up. But thank you for I think the three words that I could use to summarize your talk: it takes a village. Yeah, and so this is one of the main things. And the global burden of disease. Go ahead, Vigila. Global burden of disease has been a major issue that oftentimes it gets neglected. So thanks very much for pointing that out. Thanks. Thank you. So I met Vigila and Vicus, but maybe just right when we started, our very first, and you were one of the first few people that we met and connected right away. Been very supportive. I really thank you for all, a lot of the support and brain power that you provided to us over the years. But also, I'd like to hear a little bit more about the work that you're doing at your center. And now that with Vicus coming on board, the duo, the magic, the power of the duo will come to bear term. So thank you very much.
Well, thank you, Marco. I wanted to first thank you. Instead, you thanked me. So that's how it started. But really, all of us here should be very grateful, because Marco has put in enormous time and effort to get this together. And when he first asked me about talking during this piece of the day, I'm like, I don't exactly fit in, because I don't know that much about sarcomas. But I'm really grateful that you convinced me to talk here, because I have learned a lot. And Bill, I tried to do a little bit of research on sarcoma before, but I'm glad that you caught me up very quickly. Otherwise, I would have spent hours trying to get there. So let's see if I can make this work. So the title that Marco suggested to me was Repurposed Drug Trials, Challenges and Opportunities. And that's because we have been involved not only in finding ideas for clinical trials, but we have also tried to actually get a few clinical trials going since 2019 at Emory University. And like Claire said to you before, and Claire and I have a lot of conversations about this before, there are challenges you do not think about, even with simple trials, because the repurposed drug trials should be simpler, but there are so many challenges that we have learned over just the last 2-3 years. And so I said, okay, I will give my shot at seeing if I can talk about some of the things we have learned. So I'm the co-founder and CEO of a small nonprofit called Global Cures. And Global Cures has been around since 2007. And it is very interesting that Cures Within Reach and Anti-Cancer Fund, the two other organizations that you have heard from in the last 2 years, also came about around the same time. And the three of these organizations were, I think, the first few to focus just on repurposed drugs. So that was an interesting thing, that we have come a long way from that time in point. And I'm really super excited that we have a lot new members, new kids on the block that are here today, and they're going to be really instrumental in taking this project much further than we could at the time we started, because of the technology advances. And we've heard a lot about that yesterday during David's talk and the UDN Foundation. So there is a lot in XCure's talk today as well. So lots of very exciting things going on. When we started really to help individual patients who had no other options. These were cancer patients who were looking for something other than standard of care in clinical trials, because they either had completed the standard of care or they were not eligible for any clinical trials. And so they were looking for any promising therapies out there. And we started on this mission because I had a very close friend, 34-year-old female colleague of mine, who was diagnosed with recurrence of breast cancer. And she came to me because she knew my husband worked in cancer research. And because of my Indian background, she said, oh, there's nothing here that they're offering me. Do you have anything in the Indian, medicine, the Ayurvedic medicine or homeopathic medicine? Or does your husband know of anything? And I'm like, I don't know, but we will start looking there. And I started looking around. I have a background in epidemiology, and PubMed had just become available to us, the lay people. And so I started going through PubMed and finding interesting preclinical or very early clinical data, case studies or retrospective studies on repurposed drugs. And every time I took these back to Vikas and say, hey, what do you think about this? Do you think it would be helpful? He would say, well, I'm sorry to tell you, but this is very early in the clinical development of drugs. And I'm afraid none of these are going to help your friend, because she doesn't have that kind of time to wait. And so that's why we started Global Cures, because we had started doing this research and we wanted to be able to help other patients who are looking for these opportunities. Our ultimate goal, even early on, was to try to find money to fund clinical trials. And that's mainly because we were very naive. We didn't know what was involved. We didn't know how much money would be involved, because we said, okay, it's a repurposed drug, it's a cheap drug, the trial won't cost much. Well, I don't have to tell this group that we were completely wrong. And so about 6 years ago, we had an opportunity to move to Emory. And that point, we had decided that we needed a much bigger sandbox to get this to the next step. And so at Emory University, we were able to actually establish the Morningside Center for Innovative and Affordable Medicine with a substantial grant from a person we actually knew back in Boston, but he came with us to Emory to establish this entity for us. And with the help of that funding and the experience we had gained, that we actually were able to go much further. And there are about 4 things that the Morningside Center has now made it their priority to do. The very first thing we realized that we do need to find these opportunities. And all that work I did looking through PubMed for hours and hours and hours, I'm going to retire from that. I told this to David yesterday, because he and other people in this room are going to now help us find these. But nevertheless, when we started at Emory, we had all this data collected and all these research papers that literally sat on our computers at home. And so we have made an effort to put it online. This database will be available to the general public. And I'm going to try to show you just one of the pages. I know it's going to be probably hard for most of you to read, but basically we had worked with anti-cancer folks from Belgium to actually look at all the drugs that could be repurposed. So we had a list of about 250 drugs. This was almost 8-9 years back, so it was quite a while back. But the patients who were calling us were not calling us saying, hey, can I use metformin for my disease? They were saying, I have pancreatic cancer, stage 4, I have failed all my current therapies. Do you have anything to offer to us? So this database was designed with that in mind. We could search it by disease, and I'm not sure you can read the top line. We also were focusing first when patients called us, not necessarily on the lab data or the preclinical data, but was there any human data? Because these patients were going to probably not have that much time to wait for that lab data to mature. So are there any human studies? So we looked at, I don't know if you can see, but there are these 3 tabs here. And the 3 tabs will actually aggregate the data by retrospective studies, small clinical trials, and then case reports. So you could click on each tab for each disease and then go down that list. So again, a lot of information here, and we will be publishing this pretty shortly. And with regards to this database, I have to thank my colleagues who are here with me, Krista, Charan, and Selvi at the back, and Grishma, who was not able to join us. But they have again put a lot of efforts in getting this data ready. And again, we did it the old-fashioned way, brute force, and now we are hoping that there is much better way to do this. The second thing that Morningside Center is focusing on is, of course, running the clinical trials. And again, like Claire was explaining before, our goal also was to do small pilot studies and heavily biomarker-driven studies whenever we could, because we do, these trials are getting done at an academic health center with all its infrastructure for biomarkers. And that we are hoping would allow us not to just do single drug trials, but might give us some clues as to how to combine drugs. And I won't go through all of them. The very first trial is a trial that we have talked about for many years now. And this is in the pre-surgical setting with a goal. The goal of that trial is a pilot study of biomarker evaluation and safety for pre-incisional ketorolac. So these patients who are undergoing curative resection for their non-small cell lung cancer or renal cancer would receive pre-surgical ketorolac. And there was small data out there to suggest that this would actually reduce their recurrence rates. So we decided that we would try to do the study and do heavy biomarkers with it to see that if the ketorolac, the subjects who received ketorolac actually had something in their blood that could tell us whether they were getting an immune response to the therapy because of this intervention. And I won't go into the details of why we thought to do this, but this study has now completely enrolled for the renal cell carcinoma, and we are waiting to get the data analysis done. The next one is paracalcitrol, which is a form of vitamin D and hydroxychloroquine. You've heard Claire talk about the hydroxychloroquine study as well, and we have tried to combine it with vitamin D in pancreatic cancer, because again, pancreatic cancer has very heavy-duty stromal components around it, and many times regular drugs do not even reach the stroma, and we are hoping that paracalcitrol will help actually get the T cells to infiltrate the tumor, and then hopefully hydroxychloroquine by inhibiting autophagy will help with some of the chemotherapy effects. And that trial, we had an interesting problem with that trial, is that our investigator changed jobs right in between. And so it took us a few more months actually to get the new investigator ready to take the trial on. So it has been slower than we expected, but nevertheless, we do have at least a few patients enrolled on that trial. The next 2 trials are actually beta blockers, propranolol in particular, combined with pembrolizumab, which is a checkpoint inhibitor. And this is in urothelial carcinoma. And the next one would be the same drug combination, but in melanoma. The melanoma trial is interesting in the respect that we all understand that any given medical center may only have so many patients. And enrolling patients fast is something we wanted to be able to do. So we are also on the lookout for other organizations who already have the trial started And if a PI at Emory University is interested in joining that trial and enrolling for that trial, we are reaching out to these folks to see if we can actually get these trials done faster. The neoadjuvant simvastatin and letrozole in early stage breast cancer, that's the new trial that we have just started. And the last trial, the treatment of brain metastases in arginine supplementation is also just about ready for enrollment. And we also have a few other studies that are in clinical trial development, and hopefully we will be able to get them started quickly enough. As everybody in this room is probably aware of, clinical trials kind of took a little bit of hit during the last 2.5 years. Because with COVID, patients were afraid to come in, and especially if we needed them to come in several times for the trial, that was a problem. And we had actually clinicians were burnt out, and the clinical trial coordinators have been in short supply as well. So the trials should have started, but have not yet started enrolling. So with that explanation of what we are doing, we also do a lot of education and advocacy as part of Morningside Center. But I wanted to, in interest of time, actually go to what are some of the lessons we have learned. And again, some of these, when I started putting them together, they seem like obvious things. And I think many people in this room are going to find these things that we find as challenges very obvious things. But again, when we started, we were very naive and we didn't know about these simple things that would kind of stand in a way. So the very first thing that we thought, when we began this whole enterprise, we thought funding would be the limiting factor. And we have all talked about how funding is critically important. But we tried to start the trials, actually getting a PI was problematic. And why was that? So if you look at this diagram, these are the major tasks somebody has to do in order to do a clinical trial. And all pharma-funded trials have two entities that are working together. The principal investigator is one of them, but the pharma who is the sponsor of the trial actually is involved in many of the tasks that have to be done, which are very time-consuming. And when you have repurposed drug trials, they're mainly PI-initiated trials because we don't have a pharma as a sponsor. So if that's the case, then who does all the tasks that are assigned to the sponsors in regular trials? And that is the same person. The principal investigator ends up being the sponsor and the one actually conducting the trial. So they end up doing all the tasks that the sponsor has to do. And many people have told us that if, especially at an academic health center, you have a drug company-sponsored trial coming in, the PIs would rather go on that trial because they have all this support that they will not have on a smaller repurposed drug trial. The other major thing we found that most of the investigators who were interested in doing our trials were actually young investigators, right? Because they were the ones who were wanting to get into the clinical trial arena. They wanted to learn. So these really were folks that needed a lot of mentoring. And really, there is no formal training available in clinical investigation for most physicians who are just MDs. It's not taught that much in the medical school, if you will, because most of them won't ever need to be involved in clinical trials, those clinicians who are just treating patients. So when we wanted to enroll start the clinical trials, we were having a little bit difficulty with this situation. So we had to find people to handhold the PIs. And again, my colleagues at the back there, Krista Charan has been very knowledgeable about how to help these clinicians to actually get a trial going. The budgets for the trials. And Claire mentioned in her talk that most of these trials are shoestring budgets. In fact, I was talking with her the other day, and Cures Within Reach trials are about 50 to 100K at the most. So that's actually a small budget for a trial, because especially if you want to do multi-center trials, because just getting a trial up and running and opened at one center, we realized could cost as much as $10,000 to $15,000 before you have even done anything for the patient. So the trial budgets are pretty small. And so if we wanted to add biomarkers, that actually takes the budget much higher. And so that was something that clinicians told us, that when they had such small amount of funding that they were going to apply for either through the Morningside Center or going somewhere else like NIH for the grants, the salaries that were covered for them, where it was a very small portion of their salaries were covered. And I translated that to about 2 to 3% of their salary coverage, which is about 40 to 60 hours. And from the previous slide, I told you these clinicians were doing a lot more work than they would do for a pharma-sponsored trial. So of course, I'm not probably going to spend too much time on this. I know we are running a little bit late, and we have actually talked about funding issues in the last 2 days as well as today. But one particular point that I wanted to bring to your attention, and this is something we have been encountering all the time, is when you go to any organization asking them for funding, there are reviewers who review your grants. And if you compare the kind of data that is presented as a rationale for a repurposed drug trial, that data package actually looks somewhat different from the data package that a pharmaceutical company would be presenting. Pharmaceutical companies have a lot more rigorous preclinical data. They have much better handle on the mechanism of action, whereas a lot of repurposed drugs we are finding are actually pleiotropic agents. They have many different mechanisms of action they could be hitting. We don't exactly know which one of the mechanisms might be at play. Metformin, I'm sure most of you have heard that metformin has been tried in many different cancer types. And we're not really sure which one of the pathways it might be affecting there. So that's one of the problems for reviewers, that we don't necessarily know that, this is the mechanism of action that might be of value in this particular trial. The dose response, the PK/PD, we don't usually have that. Maybe if you're going to use the same drug regimen, we do have some of that data available from the label, but if you're going to use less or more, we don't always know what that data is not well defined, in other words. On the other hand, the last row is important because a new drug has never been tested in humans, whereas most of the repurposed drug data actually is coming from human use. So we have, as I mentioned, case reports, retrospective studies, and some very small trials that have been already conducted. So because of this difference, there is, and there is no other easier way for me to explain it, but there is a general bias. And I know that you folks at CDRC and FDA, we have heard this on our meetings many times. Some people are very honest about it. They said, well, we don't think repurposed drugs are going to work. And that is their bias based on what I'm not exactly sure, but I think the data package may have something to do with it because they just believe that the data may be inferior that we are presenting for the repurposed drug studies. And again, I've already touched on this topic that most of the trials that we are funding are small trials. But there is another aspect of it that actually, when I used to first look at all these studies that were being done, I would always say to myself, why didn't they do this or why didn't they do that? That would be an obvious thing to do. And now I know the answer to that. Their funding is limited, so they had to make a lot of concessions. So yes, they wanted to include a comparator group, but they only had funding. So they said, okay, let's just do a small single arm trial. And that's why many of these clinical trials that are actually already finished in repurposed drugs have a lot of gaps and they're not optimized really, that the design of the trial may not be optimum. And inadequate funding is one of the reasons why that is the case. The third thing that we found, which was also interesting and we didn't really think about it when we were starting these trials, is many of the repurposed drug trials actually do not finish. They just are not, there is no successful completion of the trial. And I just literally put metformin and cancer in the clinicaltrials.gov. And some of the examples here will show you that these trials did not finish for two major reasons. One of them is that there was no recruitment. because as I mentioned before, we have young investigators doing these trials, and recruitment is not something, it's a big piece of running a trial, as you all know here. We didn't think about that before, but if you can't recruit patients, the trial eventually will stop. And the other big piece, we experienced ourselves that clinicians who start the trials, they get new jobs and they move on and the trial comes to an end. So those are the two main reasons why many of these trials actually are never completed. So these are the three different reasons why clinical trials face challenges with repurposed drugs. And I've taken one shot at trying to say, is there something we can do about some of these challenges? So the recruitment of clinical investigators, basically, we really need somebody to do the training and mentorship for these young investigators who are interested in running repurposed drug trials. And many academic health centers have infrastructure that they can get it going, but maybe having a more universal way of training these people might be useful. And if we had such a training available, maybe it would also be useful for pharma-run trials. So there could be something that we could actually leverage for pharma trials as well. Because basic problem is that we need, the other major problem is we need a sponsor for repurposed drugs. And so a national agency, the talk that Vikas gave on Tuesday, might actually fulfill all the steps necessary for recruitment of clinical investigators. And the same thing is probably true for the successful completion of the trial, that if we had such an agency, they would look at different trial designs and they would help recruit patients on a national basis. They might even help us actually get more centers opened all at once to get the trials done more quickly with higher enrollment. And in terms of the last one, funding, what Cures Within Reach and to some extent Global Cures and Anti-Cancer Fund have tried to do is we have tried to focus funding on just repurposed drugs. Because for the reason I mentioned before, this bias that we have, if you put all repurposed drug grants in the same group as other regular grants, they may not get funded. And we saw that with Active 3, we have some information, some underground information as to why there was no repurposed drug for COVID in Active 3, because they felt that, okay, if we have a pharma trial, we are most likely to have successful trials. We're likely to have funding for the trial that would be better, and so on and so forth. So it is possible, and it may not be completely unreasonable to say, okay, we just need a completely different funding track for repurposed drug trials. And this is my last slide. So what is the opportunity? Instead of going through many different opportunities, I think we have an opportunity for this group. I don't have to sell this, but we really are at a point where we need to think about other ways of creating new treatments that are fast. We don't have years and years to wait to get new treatments for many of the patients who are right now waiting, and some of them have not too much time to wait either. We want them affordable, because many of the treatments that are being that are coming in the news these days have a label of like $100,000, $200,000, $300,000 per patient. Even if we might be able to afford it, I don't think we can afford it here in the US, but the rest of the world definitely cannot afford those treatments. And so we have an opportunity to rapidly develop new, effective, and affordable treatments for unmet medical needs. Thank you.
Thanks, Rachel. So while we're getting settled down, what I thought I would do, I was actually going to have a good 15-minute presentation, but we're running so late. I'm not really going to spend a lot of time on a few slides that I was thinking of doing. So before we go to the panel discussion, and here are the panelists, and we'll be inviting them on the stage shortly, and we'll ask them to make their introductions as they come up on stage. You've seen this slide earlier today. I think Bill showed it about the challenges of the diversity of sarcomas. And then also, as we started taking a look at this, and this is actually last year or 2 years ago, I think, Bill, you gave this presentation, and I took a picture of it because I'm like, you know what, this is sort of all the things that we need. There are all these different mechanisms that we actually know now, but nobody's really utilizing that. And then what is the repurposed drug that can be used for affecting these pathways? And so getting more into repurposed medicine or personalized medicine. What is the genomic? What is the epigenetic issues that are there? What are the transcriptomic perturbations? This is going back to what Bill was talking about, this sort of overall ecosystem of doing this. And then, can we capture real world data that's being generated every day regarding repurposed drugs? These are as we start talking about different patients and patient groups, these efforts are coming to light, whether it's checkpoint inhibitors, whether it's for some of the more general soft tissue sarcomas, such as this second generation BM1 inhibitor, but that's only designated in EMA. So, potentially something in the US that we could look at. Potentially, I think that there is an opportunity here to explore it a little bit further because we know the mechanism there. So let's take a look at that pathway in several different sarcomas and see if that's a way of looking at it. And so I'm thinking about this more of the, there are opportunities and we don't know how far down the road we can take those, but are there some gold nuggets there that we can start taking a look at? And here are just some four examples that we thought about. And the one that is example number 2 that we're really trying to go after is the rapamycin used in PCOMA, but there are other opportunities here as well. And so, what's in the pipeline? And is there sufficient data on the off-label use so that we can start making an analysis? And that's a big question. But I think that with the molecular revolution over the past, I would say 5 years or so, the price of genomic sequencing is coming down. A lot more people are doing it. It's going to be a utility tool, but can we use those molecular markers to identify genomic correlates of response? I think that's something that we can do. We're trying to engage many stakeholders. So there's a lot of stakeholders in this room. And as we start having these conversations of who's here and who's not, most importantly, who's not. And those are the red boxes here. We've not been very successful other than NHS outside the country of having a payer. But within the US, we've not been very successful. I haven't really pushed it either. So it's something that I think over the next year, I would like to explore this a little bit further, whether it's a national group or whether it's private insurers, for example, that may be an opportunity. We haven't explored that yet, but it's something that I plan on doing. Industry has always been a challenge, but at least when we have spoken with industry, they just said, keep us up to date, keep us informed of what's going on. There may be opportunities a little bit later on, and they're not completely devoid of that either. Funders and clinical trialists, we're starting to get them on board, but the rest of them, it's been a really good conversation and discussion. So we started this a couple of years ago. Unfortunately, it was right in the middle of a pandemic. So there was a lot of sort of juggling about what we needed to do, what was important at the time. And one of the questions that I asked FDA, this is the Oncology Centers of Excellence, is, I can pick whatever diseases that we want, but what's really going to be helpful for you? What is it that you're seeing that you would want to say? And they told us 3 diseases: pleuropulmonary blastoma, adenocortical carcinoma, and sarcoma. So those are the 3 diseases. And so I went through and took a look and asked some specific questions about, what are those diseases? Are there clinical trials? Is there a company that's doing it? How many clinical trials have been done in the past? Is there drug repurposing occurring? What's the mechanism of action? So these types of very basic questions to try to figure out. And that's when I met Bill and Denise. Unfortunately, Bill, that part of it wasn't really to find out what was going on. It was great, but it was more of an interview type of section, because I wanted to find out who would be great to work with at the time, because I knew that if we were going to move forward, I'd have to work with you, Denise, or whoever it was that was going to be. And I just clicked as soon as you came on, and I'm like, yeah, you get it. I know what you're doing. Denise was an absolute, an angel for me, because to try to identify the various different patient groups and to bring them on board was going to be, I think, if without her, it would have been very, very difficult for me to do. And then once we identified sarcoma, then we sort of said, okay, there are 77 rare sarcomas. We're not going to do all of them at once. So then we had to just go through this process of identifying, well, out of all of those, what's going to be something that we're going to be moving forward with. And then after several rounds of discussion, including with you and with others, it's, you know, the angiosarcoma, the PComa, and epithelioid endothelial sarcoma. Getting the stakeholders together, getting sort of the identification of what is off-label and what is not. And then we took a lot of time to try to figure out what is the case report, what is it that we're trying to capture. So we worked with clinicians, we worked a little bit with the patient group. So it was really more of the clinical piece that we were trying to get. What is the clinical area? And over the past 6 months or so, we've gotten it down to a much more reasonable amount. But then we've been having, over the past 2 or 3 months, conversations with the Oncology Center of Excellence, tweaking those responses, and then working with NCATs, who are going to be taking those and developing a case report form that's in electronic form. And thanks so much for Heather to help wrangle those discussions. But I think that over the next few weeks, we'll have that. And so we have now explored partnerships with defining a process of securing data. So one of the questions that I asked the clinicians, how do you want to do this? You know, do you want me to send you a form and you fill it out? Absolutely not. Who's going to want to do that? So they said, just go to our institution and do that. So rather than doing that, I started to take a look around to see who else is doing this? What other partners can we explore? And that's when XCures came. There's a number of other groups that are doing this as well. So had some conversations, worked through some of these issues, but eventually we came to the conclusion that XCures is a group that we really wanted to work with. So developing legal agreements, setting master service agreement, and then figuring out how to secure data. That's sort of where we are right now. So over the past few months, we've completed a stakeholder review of the clinical case report form. We're now working with our partner NCATs to develop that app. We think that it'll be done in around November timeframe. In about a month from now, we'll start a data collection, I think. And that's not saying that we'll have data in a month, but we'll be starting the engagement with clinicians, with patients, starting to go down that road of identifying patients. About a month after that, we want to also start taking a look at what that patient case report form is looking like. This is one of the main things that when we discuss with patients, it's like they have some data that won't be in the electronic medical records. And that's the one thing that XCures has always said, hey, I can only get you what's in the medical record. If it's not in there, I can't get it. So there's going to be a lot of information I think will be important there. And it's also a way to sort of correlate whether, you know, what's in the medical record is actually matching what is not. And what's in from the patient. And so we'll have an opportunity to do some patient collection. And by the time we get to about the beginning of next year, I'd like to sort of take a quick assessment. Where are we? How are we doing? And then see if we need to make any adjustments there, because we'll have another 6 months or so after that to sort of make some adjustments and get some data to do some analysis. So this is my map, at least how I'm thinking of it as we're moving forward. So, you know, our first stage is sort of this education campaign. Who are we? What are we doing? Why are we doing this? How are we doing it? What are some of the security issues that are coming about on this? And really taking those to both the clinicians and patient advocates and helping us to identify who those patients are, to then have those referrals and have a campaign for patients to say, this is what we're doing. We need your help. We need you to do this. And it's basically going to the XCures platform. And initially it's for PComa, where there is an existing informed consent, an existing IRB, existing protocol. And this is hopefully where Denise's group, EHE, will also come in through XCures. And they will be able to secure data from through third-party right of access to grab these types of EHRs, insurance claims, the labs, the images, and potentially even PROs. And that's the essential data that we'll need eventually to, I think, link in to what Bill's presentation was about, what else can we do about this, and the ability to collaborate more effectively to design better clinical trials. But from our perspective, this is a very slim design number of data elements that were defined in that earlier stage to have that clinical case report form being made available to the CURE ID app. And that's at the FDA. So another aspect of this is that in having discussions with Corey Painter at the Broad Institute, she has roughly about 62 patients for angiosarcoma already done, curated manual extraction and made available. So through their CBio portal, I've got a couple of fellows at FDA working through that, pulling that data together and seeing how well that works. So that's our first cut at how well does that case report form works. In another 2 or 3 months, we hope to have a patient case report form very similar to that, but then having that ePro action. And we want to be able to talk with both the foundations that are doing these types of questions. So we don't want to be able to repeat, hey, how much can we actually learn from what you guys have done? What's acceptable, what's feasible, and make sure that we're not duplicating. So we'll work through all of those issues, and then eventually have that go into an open access CURE platform. So it'll be an open access platform, very limited data de-identified, but also this other platform at XCures where the clinicians can really have a more of a direct access to a particular types of questions that they have with regards to patient access for designing clinical trials. So I think that is it. I'm going to call the panelists that we've identified up to the stage, and we would like to start having a few discussions. So we had actually a call about a month ago, no, actually a couple of weeks ago, and had some discussions about some of the topics that we'll be covering. So we'll go through some of those. But also I want this to be more of an opportunity to have discussions with the audience. So if people have questions, feel free to raise your hand. We'll have a couple of microphones that we can pass around, and then feel free to do that. I think this is the first time we had actually an all-female panel today, right? Yay. We can turn that off.
Thank you.
So maybe we can go, the first thing that I thought I would do is just have an introduction, just go down the row of who you are, where you work for, and sort of how you want to, what's your interaction with regards to drug repurposing? Like, why are you here? Why is it that you're interested in drug repurposing, especially for oncology or for rare diseases?
Yes. All right, I guess I'm starting. Hi there. Thank you so much. Really excited for this opportunity. My name is Andrea Gross. I work at the National Cancer Institute in the Pediatric Oncology branch, particularly focused on rare diseases and tumor predisposition syndromes. So a lot of my work is focused on neurofibromatosis, which is a genetic tumor predisposition syndrome. Patients get a variety of malignant and non-malignant tumors. One of the medications that we've used for that is a MEK inhibitor, which was originally designed as an anti-cancer agent, and has been, even though it was not approved previously, was really repurposed, frankly, for this kind of non-malignant indication. And so that's where kind of I come into this. But in addition to that, I also work with the broader RASopathies community, which is other syndromes like NF that are caused by mutations in the RAS pathway. And we're really interested in how can we use existing medications, particularly those that were potentially originally designed to target cancers that have this abnormality that might also help these patients in a variety of other ways. So that's where I'm coming from.
Yeah, and this is actually a really good segue that we also had a very keen interest in other rare diseases, and one of those rare diseases actually was ursopathies, and so we've connected quite well with the NCI to be able to do that. Had very good partnerships, and again, it's these MEK inhibitors for, although they're being used for what they're being used for, there are these rare ursopathies that we really want to be able to, and nobody's going to do that. There's just too few patients to be able to do it.
So hi, everyone. This has been a really amazing 3 days. So thank you for having me. My name's Christine Heskey. I'm also a pediatric oncologist, and I also work at the pediatric oncology branch at NCI, but a little bit different from Andrea. So I'm a bench scientist. So I run a translational lab looking for potentially new therapies that we can bring to patients specifically with pediatric type sarcomas. And then I also run a clinical trial kind of arm of my research program where my goal is to take the most promising things that come out of the lab into the clinic. And I think Claire had a slide earlier with her definitions, and she had something called rescue drugs. And I feel like I live and fail in the rescue drug space because in pediatrics, very often the timeline is lagging behind what's going into the adult oncology investigational space. And so I have been on the unfortunate end of several trials where we're waiting for a drug company to give us access to drug for a pediatric study. We finally got it, and then the drug failed in the adult indication, and they pulled the drug, and we had to close the study, or we couldn't open the study for that reason. And so I'm very interested in repurposing. I actually work on the bench side with NCATs, looking at data from drug screens using repurposing drugs. And I'm just in this struggle space trying to move those things into the clinic.
So I'm Vidula Sukhatme, and I've actually spent last 15-20 minutes talking to you about what I do. So I won't go into any more details on that, but I do want to say something that I wanted to do at the end of my talk and I forgot. Not only Marco, you have spent a lot of hours, but we have had a lot of conversations with Chandler and Kitty and Jennifer and all your staff. So I want to thank everybody for doing a really wonderful job of getting this group together.
My name is Brandy Felser, and I'm from the Sarcoma Foundation of America. And we are the largest private funder of research in the sarcoma community. And we also are kind of pan-sarcoma, so we're not subtype specific. We also are very comprehensive in that not only do we fund research, we also provide education and patient support, and we also do public policy advocacy as we are based in this area. So when you're based in this area, it's really easy to do that kind of thing. So we are involved in drug repurposing, obviously from a funder standpoint. We do have people reach out to us asking us for funding for different research projects that they're doing or clinical trials that they're interested in. And also, if you fund research, you want to bring treatments to the market, right? What's the point of funding research if you're not going to see any results from it? So as a strategic priority for our organization, it's that through line of preclinical all the way on through. And then how do we meaningfully engage the patients in that and also empower the patients to be good advocates for their care and accrue trials. That is something that's extraordinarily important for us because, again, if we're going to fund research, it's really important to get the patients in the trials or we're wasting our time. And I think that, again, from a funding standpoint, it is important for us. I think, especially in sarcoma, everybody's done great presentations. You've heard a lot about it today and got kind of the Cliff Notes version of sarcoma. But if we're really going to bring treatments to patients, because with so many subtypes, I think that's the greatest challenge for us is A, time is of the essence, but B, from my standpoint, it's overwhelming because we have so many subtypes that we focus on as an organization and we don't have a lot of patients. So how do we get those treatments for patients in a more meaningful and a more faster way? And repurposing is one way, really interesting way to do that. So that's me.
Thank you. Nice to see you too in person. So I'm Lenny Woods. I'm co-founder of the Clear Cell Sarcoma Foundation. My daughter was diagnosed with clear cell sarcoma in 2016. Tumor removed, clear margins, small, less than 2 centimeters, maybe less than 1, GI version. We thought we had won the lottery and we were good to go, and we were very thankful. 18 months later, it came back in the same spot. It had infiltrated, 5 lymph nodes active for cancer and a greater than 5 centimeter tumor. We had done enough research to know what that meant. That was in 2018, 5 years ago. We've done nothing but surgery, and she's doing well. And the reason that we started the foundation was because we're an anomaly. Jill, who is my partner over here, she too has a daughter who had a similar outcome experience, but that's not typical for our patient group. Also, we have patients who are diagnosed, have the tumor removed 15, 20, 50 years later. The tumors come back and take their life. So it's, we're up against the time. We're in a race against time for our life. So I'm interested in repurposed drug testing because the chance of my daughter having a long life are much better with the drugs that are already out there than trying to create one from scratch. So we've also done some work getting together. Pharma has been great to us. They helped support a phase 1 trial. As you know, I wouldn't even say recruitment is hard. Retention in a phase 1 is hard because they go in at the end of life. So it took a lot longer than we thought. But I'm really thankful for Marco bringing us together and Denise for thinking of us to be one of the groups. And I appreciate everybody here today.
Hi, everyone. I'm Sue Anna Brunicky, and I'm at the American Society of Clinical Oncology. And we have a phase 2 signal finding study called the TAPER study that is active in the US. We have 251 sites. It is a biomarker-driven trial. So it's repurposing because we're taking FDA-approved drugs and looking at their uses in other cancer types that are also biomarker-driven. In our trial, we form cohorts that are based on the biomarker, the drug, and the cancer type. Some of the, in cases where we have biomarkers that are really rare, we also will, put all the cancer types into one cohort. We have a sort of an early stopping rule at 10 patients. And then if we see a signal, we go up to 28 patients. So it's a, and we work with 9 different pharmaceutical companies and have offered over the course of time 18 different therapies in the study. So definitely, finding patients with rare cancers with rare biomarkers is really important for the study purposes. And that's really what's driven us to open a lot of sites. We work mainly with community-based sites, but we also have academic sites in the network. And then we also work a lot in collaborations with other organizations, including the FDA Oncology Centers of Excellence, on different initiatives, related to broadening eligibility criteria, making clinical trials easier to do, and more pragmatic. So that also a lot of what we've talked about over the past couple of days has been very meaningful for that reason too.
Thank you. And I also want to mention, Lenny, thank you for attending, because, this is a challenge that we have, although we can only, identify 2 or 3 that we can go after because we can't go after everything. What was been really inspiring is to say, you're not going after clear cell. I'm not going to hold it against you. I'm going to be mad a little bit, but yeah, exactly. But for you to actually attend, participate, and engage with us, it's been amazing. So thank you very much for doing that. I really appreciate both you and Jill for doing that. When we did have that conversation about a week or 2 ago, we came up with a few general issues, especially for just rare diseases in general. Some of them were covered here. So for example, the slow enrollment was one thing that we talked about. There were other challenges, especially for pediatric trials. Do we want to touch base on some of those, especially because some of these are very chronic? Sometimes they can be rapidly progressing, but other times it takes several years, and you go from one age group to another, and then how do you manage that? I don't know if Christine or others would want to talk about that from a pediatric standpoint.
Yeah, sure. So I think, you know, the biggest problem that I have encountered in terms of our population is not necessarily pediatric specific, and it has less to do with the patients that have the chronic disease. I think Andrea can probably speak to that more, but I work in the common, which are not very common, the common pediatric sarcomas, which are all actually very, very rapidly progressive. And I think, you know, the difficulty with that is in terms of getting patients on trials, when we have trials that are open at very few sites. Somebody said yesterday, like, it's much easier to bring the clinical trial to the patient than the patient to the clinical trial. And we just often don't have that opportunity. And so, you know, COVID obviously made a huge impact on this, because there were obviously logistical, but also kind of ethical issues of saying to families, you know, put your child who's, you know, critically ill with immunosuppression on an airplane to come to enroll in this study. I mean, it sort of felt like we can't ask people to do that unless we know this is going to work, which of course we don't. So I think that's a big issue. And I think, well, I'll let Andrea talk.
Okay, well, and along with that, that was even more so for our natural history studies that we did have. You know, we do have some natural history studies that are ongoing for some of these rare diseases, and the pandemic, I mean, we absolutely could not bring anyone, right? There's literally no direct benefit for people traveling. So like, stay home, do not come. So it really, really was challenging. So yeah, I work more in the pediatric chronic disease space with patients with tumor predisposition who can develop some of these really aggressive soft tissue sarcomas and others, but who often live with kind of more long-term, but very rare conditions. And I think one of the things you were alluding to is the transitions of care is a huge challenge. And it's why I already hunted down Dr. Shapiro, Dr. Tapp, but also Dr. Shapiro from the CUREs, and was just amazed at the possibilities of being able to get records for patients across multiple institutions without what currently happens, which is our research nurses spending a lot of time, or me or others, calling institutions and spending a lot of time, you know, I kid you not, on fax machines still, you know, which, you know, it is mind-blowing the possibilities of that, but it's a huge challenge. And our adolescent and young adult population in particular, they go from pediatric to adult. The other challenge we have is that not everyone is treated by the same type of doctor. So for example, in some of the really rare syndromes, depending on where you get diagnosed, that's sort of your home. So it might be the geneticist that treats you, or it might be the neurologist that treats you, or it might be the oncologist. But outside of oncology, not very comfortable with clinical trials. And so making sure that we get patients together from all of these different places can be a real challenge.
Bill, did you want to allude, because you alluded to the fact that you at Memorial Sloan, that you do have that capability and you've addressed that.
Yeah, I think it's challenging because we saw too the point of entry, you know, in adolescent and young adults, really people would have drastically different care even at a single institution. So one thing we tried to do is harmonize all care paradigms. But I think when we think about the transitions, it's also all of the psychosocial aspects of a young person that comes into their care and their ability to maintain care, right? And I think that's one of the things we spoke about, the psychosocial determinants of health and how we can better understand that. And I would also love to use these data sets to begin to really think about all the challenges that patients face. And it's one thing to pull them from the EHR, but if we can also get patients and their families to bring in a lot of the variables that probably could affect it. And I agree, like and all the work you're doing there and have done is amazing. And it's such a challenging patient population because they spend so much time in different offices and different healthcare offices. And, you know, it's young people wanting to be young people, and how invasive we are in trying to care from them, right? So how can we normalize that? So it's just, you know, incredible.
Very challenging. Any other thoughts or comments on that from the panel, or?
I'll just share that in our trial, we had a pediatric cohort, and we struggled with it because when you're rare and you're trying to fit rare into a trial that was set up decades ago for not rare, it's very difficult. So at any given time, we'll have several pediatric patients. One thing we run into is they often get diagnosed, we're an EWS sarcoma. So there's some other EWS sarcomas that have treatments. They'll get diagnosed with that and take a treatment. Then they come to us, the patient organization. But when we, like, for example, we had a 9 and an 11-year-old that could have gone into that trial, but the rule was you had to be 12. You know, we've had 4 between 12 and 18 before at a time, but it's at any given time, you almost have to have the trial open for 10 years to be able to get the right amount of people. So we have to change that. And I don't know how, but y'all are shaking your heads, and you know, I am very thankful for both of you. Help me fix it.
That issue of not having the trial open at the right time and the right place for the right patient is one of the things that all the discussions about decentralized trials has been so fascinating. We do, to some extent, utilize like our home providers at the NIH. So when we do clinical trials at the NIH, people are able to get their physical, some of the interim visits locally, but they still have to fly out to get the drug. And like you said, you know, the challenges of you have a patient and the eligibility criteria. I've heard that multiple times over the last couple of days, and I feel for that so much. But I don't know how to fix that, because when the prospective clinical trial, there are some things that you have to balance, right? You want to make it as broad as possible so that the most people can potentially benefit, but narrow enough that you get information that is generalizable and useful. And I'd love to hear thoughts from the smart people in this room about how to make that better.
Yeah, so if you have ideas, thoughts, questions, other things that you want to raise in the audience, please raise your hand. We'll get you a mic.
In our trial, we do allow patients as young as 12 or 14 to participate, but that's because it's a biomarker driven trial. And so, you know, they're really, I think the hypothesis is that biomarker is going to react the same, at least, you know, when someone is sort of post-adolescence. But that has been challenging, because even dealing with drugs that are already FDA approved, sometimes the companies are reluctant, you know, for us to use them in a under 18 age population. Right. And we do at least require that there's at least initial information to establish a pediatric dose. So there has to be some, you know, pre-existing work that makes us comfortable about at least knowing how to dose the drug. But
so that you do have some PK/PD data to be able to tell the sponsor that is giving the drug. At least we know roughly what the concentration will be, and we'll be monitoring for that, right? And then you mentioned also getting the drug to the patient, even if it was a decentralized trial. And so, can you just elaborate on that?
Well, I'm just speaking from my own experience with, for example, the MEK inhibitor trial. For a long time, shipping drug, right? Are you allowed to ship a drug? Well, if it's an investigational agent, you may not be allowed to do that, depending on the sponsor of the study. And so there was actually one point when we were talking about, we do pay for transportation for patients. And basically we were talking about bringing not the patient, but their parents from California to DC to pick up drug and fly back because we weren't allowed to UPS it. And it was crazy. And we're like, this is not a good use of the taxpayers' dollars. This is not a good use of anyone's time. You know, that person has to take off work. And so thinking about how to, now thankfully that has been improved in part, I think, with the pandemic. Actually, some of the rules were alleviated, but it's a challenge.
Yeah, I was just gonna shout out to the regulators, because I'm not sure if everyone in the room knows, but the reason that 12 is this number is because there are some data, and it doesn't have to be a biomarker driven study. It can be a study where there's some evidence somewhere that a patient would be benefited potentially by this therapy. And the FDA has said, you know, you can go down to 12, you don't have to cut off at 18. And I have had a lot of experience with drug companies who actually don't realize this, But if we can go to them and say, well, look, here's a statement from the FDA, that goes a really, really long way in sort of allowing us to enroll that population.
They put out a guidance document on that, which has
Exactly.
helped a lot.
Yeah. And they have some guidance documents also. I mean, I feel like someone from the FDA should be saying this, but about eligibility criteria in general, like, should we be, no, we shouldn't exclude everybody with hepatitis. We shouldn't exclude everybody with HIV if it's under control. We shouldn't always exclude people with brain mets. And so I just think like a lot of that is helpful if it can be in black and white from the regulator. Yep.
I just wanted to share like our experience when we were setting up XCures. We thought a lot about this problem, and we definitely haven't figured it out. I don't know that anybody has figured out this problem, but in that early sort of fact-finding process, we talked to some of the folks at Novartis and at Ultragenyx who had had success using compassionate use programs for label expansion. And that was informative for us because we think, all cancers are rare these days, right? They're all rare diseases. And so we were looking at the compassionate use programs overcome one of the challenges, which is we can get them started quickly. Like we had a patient at Stanford last summer, infant leukemia, relapsed, and worked with the physician there to open an emergency IND. And that patient actually was able to get a menin inhibitor, had a response, and then was able to go to transplant after that. So, it was the only way to move quickly here. Like I have not seen a trial open quickly outside of that sort of compassionate use emergency IND setting. But if you can gather high quality data, you could sort of aggregate and generalize these. And I think that's sort of a pragmatic approach for some of these things, because otherwise you just can't open a trial out at a community site with an investigational agent quickly enough. Just my 2 cents.
It's a good point, but I think one of the things we should be talking about here is how to simplify the process, right? Because we're asking a different question with drugs that are often available. And sometimes, we have reasonable data sets already out there, and we all know the clinical trial process is way too cumbersome for reasons that no one can explain. And if we have the experts and we have patients talking about the data sets that would be meaningful for them, and we have drugs that we know about, as was adequately said, maybe we should be simplifying the process where we don't have to follow, the same rules, redundancy, paperwork, cost, right? That's now built into the system for reasons that are not always regulatory, and say, how do we do this correctly, right? And I think that's why partnering with regulators is all important, because we may realize what are the things that, are important to do this, and what are the things that are just adding cost and craziness, right? Because we're asking different questions. We would love to know what's the usage pattern of a MEK inhibitor in neurofibromatosis across the country, right? That would be really cool. Or rapamycin is a drug that someone can write a prescription for, so it's not a drug delivery problem. It's a question of understanding outcomes. So thinking about those questions, coming to consensus within a group like this, I think could really be helpful to drive this process.
So one of the things I think, Suana, that we wanted to talk a little bit about was Citi. That they've developed some guidelines on decentralized trials and some of the challenges with that, but also how we potentially could approach those in different scenarios and situations. Again, decentralized, there are definitely some challenges on the regulatory side. We work with, I think Trevin Locke was here earlier this week from Duke Margolis, that they're working on this as well. And we got at very early days, I think back in October of last year, started engaging with them. And essentially they're seeing the same sort of thing. They sort of articulated that issue, those 3 main issues. What are the technologies to be able to enhance and be able to do decentralized trials? So electronic medical records, things like that. Can we embed trials in clinical practice? Also just decentralizing them, getting them into an area where they're being done in the community. Obviously great, but there are some barriers to that, so that all of a sudden, so that's everybody that's on this drug, if you have 15 clinicians in an area that are doing this, do all of them have to sign a 1573? These are the sort of the administrative hurdles I think that you're alluding to, that we need to be able to sort of say, I can understand that for a first in human trial that has never been FDA approved, but can we start backing away from some of those regulatory hurdles to make things a little bit easier? Mark?
I actually don't think like the 1572 is the hard part. I mean, with a lot of it is at the institutional level, and it has to do more with like negotiating a contract and legal and compliance, insurance, and those discussions take a long time. And so our success has really only been that, you know, if you can get the lawyer at the institution on the phone to say like, there's a patient who has, you know, 4 weeks to live, can we move this contract along? You can usually do that. But it's not like the 1572 is not that hard. And in compassionate use, you're still doing, you still have to follow GCP, you still have the same regulations. It's some of the other administrative stuff. I don't always think it's a regulatory issue.
And I would say, you know, with our network, we are, the TAPER study is a platform study, and it's all biomarker based. It's a single protocol, and each drug is added as an appendix. So, I mean, the nice part about it is the protocol stays the same. All of our contracts stay the same with all the sites. And whenever we add drugs, you know, it's very easy process. So I think, you know, taking advantage of a network situation like that, you know, where you can use a central IRB and, you know, have certain things. I mean, and we are now, we just submitted an application to PCORI to do a study on dose optimization with a CDK4/6 inhibitor in people over the age of 65. So, you know, again, it's a very not a high risk trial, but a trial that asks an important clinical question. So I think, you know, in those kinds of situations, and even in that situation, we had to be very rigorous about saying, only collect the data that we know we're going to use and keep it very streamlined. So I think, you know, in those kinds of situations, and that's, you know, the city project that we're involved in, that released recommendations late last year, was all focused on how can you embed research in practice? So how can you take advantage of clinical workflows that are already there, that exist, and do things that as closely as possible follow clinical care. So I think a lot of times, you know, that's what can get things gummed up in the process, is when you're doing something and you're not following exactly the same schedule. But with a lot of this stuff, and like with our TAPER study, we're telling clinicians to just follow the label, you know, use the drug as they would use the drug in clinical care. There's nothing, no testing, nothing that differs from how you would do things routinely in clinical care. And I think that does help a lot. And we also have very broad eligibility criteria. So you're not, you know, you're not forcing clinicians to think about, oh, is there going to be that one creatinine clearance thing that's going to mean that this patient isn't going to be able to participate. So it's very, you know, simple from that perspective as well, which I think helps a lot.
Can I ask a question about the TAPER study? Because if I remember correctly, you guys had an IND waiver when you started.
Yeah, it is an IND exempt study, primarily because we're the sponsor, so we aren't submitting anything to the agency, but because we're studying largely the same population that already exists for the indicated population, it is IND exempt, which also does make it very easier because there's, you know, fewer requirements from a data collection standpoint as well.
I had remembered that when you started speaking. I think that's actually a really useful thing for people. And I know OCE has been very collaborative. Like, we've got a project we've been trying to get funded, but they already granted an IND waiver for it. And not every division at the agency is so open to that. But I think in this, where you're sort of closely following the existing use, it's a really useful thing to think about when trying to plan some of these studies, because that you could probably decentralize, right? Like, as you guys have done, but traditional trial.
So I wanted to actually address a question that you raised, not necessarily for the sarcoma or the pediatric population, but you talked about patients not being eligible for trials. So one of the things that we have talked about often is the fact that if you're using a repurposed drug for the trial, A lot of the concerns that we are hearing right now are if a drug company is sponsoring the trial, there are very strict rules who you can include and exclude. But we have thought that, and again, we have not, I'm sure there are a lot of hurdles in the process, but since this is a great group to bring this to, if you have a clinical trial that is open using a repurposed drug, is there a way we can use that protocol and have other clinicians who are maybe familiar with that drug use treat anybody? And then that way the patients don't have to travel or have any other constraints coming to the original site, and yet find a way to capture the data. And that goes to the real world evidence piece. One of the biggest issues why other clinicians won't use that particular drug is they don't necessarily know the protocol or what to do or when to do it. So if you offer them the protocol that is already running for a small group of 25 patients at one center, is it possible for us to actually try to at least get some real world evidence, number one? And the second advantage would be for patients like Lenny was saying, if they're not eligible for the trial and still be able to get that. And this will only be, of course, valid for repurposed drugs.
Right.
I think it's such a fascinating concept. And it's basically what we do in pediatric oncology. All of our patients are treated as per one of the prior clinical trials, right? We all join the children's oncology group, and even though the trials are closed, we treat them as they were treated on this other trial. We use the protocol and the roadmaps and everything. So I think the one challenge I foresee with your thought about letting patients who are not eligible for the trial do that is differentiating between eligibility criteria that are for safety, as opposed to ones that were because of what you said, that the drug company just didn't want to risk, an adverse event in a patient with a brain met. But there are some eligibility criteria that, if a patient has horrible cardiac issues, they really shouldn't be getting certain medications that can cause really bad cardiac problems. So I think it would have to, and maybe we'd have to think differently and have eligibility criteria, like these are the safety eligibility criteria, and these are the, which is an interesting way to think about it, because we don't differentiate that.
I also think in a rare disease population, I think it's I would love to collect data on all these patients that are being, all of our patients are treated off label, everything's off label, and they're doing, when we get referrals, people are doing all sorts of stuff and we are not collecting any data on it. The other concern I would have about allowing patients to sort of get the protocol and do it off study is that I think it would impact enrollment of this study. And so we definitely see that in some of our bigger trials where, there's some specific caveat to the way the treatment has to be given. And so providers just say, well, I'm not going to put them on study, but I'm going to basically do the same thing with a little bit of a difference that I, prefer. And then you lose both the data and the enrollment and accrual.
I know we have thought about that particular point. However, it's always a balancing act between getting a really great quality data versus helping a patient who needs that help today. And that's always a problem.
So, I'm sorry. I had wanted to go back to something you said about the compassionate use. So our trial was based on some really good preclinical data done in a Japanese study on clear cell sarcoma. It was using a current investigational drug that's been in a lot of phase 3 trials, combined with a repurposed drug, hydroxychloroquine. And the study, so we went to the pharmaceutical company and, well, first we had first patient go to compassionate use. I started a dialogue with the pharmaceutical company, no problem, never denied a patient. They were amazing. We had 3 patients, 3 adolescents under 18 go on compassionate use, but the IRBs, I know about Project Facilitate. I tried to make that connection. The hospitals don't have the time. They don't want to mess with it. They'll flat out say, we're not going to do it. So it took 6 or more weeks to get the drug into the child. And one was already basically comatose on life support. It took too long. So the pharmaceutical company worked with us and the FDA. We came up with a plan to say, we can't keep doing this. We've got to get a good trial going. Problem on that was it was phase 1. There were no criteria. They let all translocation sarcoma in, not just clear cell. And we followed their rules. They told us what the rules are. These are the endpoints. We didn't like those. progression, what is it? Stable disease to us should be an endpoint, but nope, can't have that, which if we had that, we would think that was glorious. All these rules that we had to follow, we followed. We did everything that was said. Well, at the end of the day, there was fatigue from the institutions because they weren't getting enough patients. The patients were going in near death. And so it wasn't the study that it was based off of said early intervention, tumor burden needs to be very low. This needs to happen early. So the phase 1, they say failed. They had a DLT call or something. I got a call from the PI and the pharmaceutical CEO, and they said, we're going to stop it. So you know what we did? We're like, I don't want you to stop it, but guess what? Totally safe, no toxic issues. Let's go back to compassionate use. Now this has been done. This is what we have to do as rare disease patients, is we have to manipulate the system that we can't get out of to save our kids' lives. This is terrible. I'm listening to all of this and it's just heartbreaking.
I wanted to say something to get, also, I want to make a comment about the compassionate use. It's interesting because I just had a meeting recently with a pharma company, and one of the things they told us is they have a trial that's kind of coming, data is coming up, and they said, we are designing right now a new trial that these patients can get into because we don't want to get into compassionate use. So we're going to design this new trial to try to facilitate getting those patients into that. But I want to say something, and this goes with what Lenny said, is, we've talked a lot. I've learned a lot over these past few days, so I appreciate that. But we've talked a lot about, collecting all of this data, which I think is really important, right? And we collect it, and how do we design the trial and all that. And, the idea of decentralized trial, I mean, it's not just an idea. It has to happen, because when you talk about rare patients and we talk about first of all, the barrier, we have a small population. The barriers are significant. In sarcoma, we only have so many sarcoma centers across the country. We don't have a lot of trial sites. I'm sure that is the same for many rare cancers. So how are you going to get these people to participate? We talk about accrual being an issue. It's not that you can't reach the patients. I had someone tell me they didn't want to drive their husband an hour and a half anymore for treatment in the trial because it disrupted her life with the kid. I mean, these things are important. They sound like they're not important, but they're important to the people that we're asking to participate. So it's not just an idea to talk about. If we are actually going to do this, we have to figure this out, and it has to be the goal, because otherwise we can talk about collecting all this data and get to design the trial. We're never going to get people in it. It's not going to happen. We deal with that. We know this. We deal with it every single day with different patient stories, and you guys have heard them all. But I think that one of the things we talked about in our pre-call was, what is the goal? And one of the things was, what is the goal? Yeah, what is the goal? That has to be the goal. And the regulators and everybody has to be at the table to have that discussion, because if we don't change some of the things that we're doing, we're really wasting our time. I'm preaching to the choir, I know, but I'm passionate.
It's under Bill, when you mentioned exactly what you're going to do, that was part of it, is right. We got to make this decentralized. I have talked to Vicus, he says the same thing. I talked to Vigila, she says the same thing. So everybody's saying the same thing. And the question is that, so what is our goal? What is it that we really want to do? And when I spoke with David and with others, they're like, do we really need to change the label to do this or not? And this is a conversation we had as well. And that was, I just wanted to throw that out there. Do we really need to change the label for what we want to do? And then if we don't, how do we change clinical practice? Because essentially that's what we want to be able to do. Who do we need to partner to do that? I think it's your organization, for example. Can we, be able to show the data on foundations as well as with clinical guideline developers to be able to sort of say, this is the data. It may not be perfect, but at least it's something, as opposed to not having anything right now. It's not going to be perfect, but it's going to get us there. And the more that people will see that, they'll say, okay, well, I'll treat that as well. Again, going on that same, well, let's put them on that clinical trial sort of thing, capture that information, add more to that database.
I think it's maybe a little bit different for pediatrics, but a couple of years ago, I had to give a talk, and I looked up the last 1 year of label approvals for pediatric oncology, and I made like a word map. And then I looked up for adult medical oncology. And then I looked up all of the approvals for pediatric sarcomas ever. And there were 3 drugs, 3 drugs, and like 2 were chemo, like some, chemo from the 70s. And so, I was the person who asked that question the first day. I think for our population, I don't think we need a label change because we already completely, basically, no offense, FDA, but like we already completely operate off-label almost
Anyway,
completely.
yeah. All of
And-
pediatrics, all of neonatology
Yeah,
is
exactly.
completely 95 to
Right.
almost 100% off label.
And there's not actually issues getting providers to kind of know what the standard is, because in oncology there's big consortia and people can kind of go to that website and figure out how to treat X, Y, or Z. And so from our perspective, I think if it's just a lot of work to have the label change and there's really no benefit and we've been operating without it for so long, I think it may not be necessary, but medical oncology could be different.
Have you explored working with NICHD for the BPCA for Breast Fractures in Children's Act?
Well, it's so funny that you say that because
Okay.
we didn't even know about them until Tuesday.
Okay.
So now we will.
Now we will. Okay.
We were looking at each other being like, wait, they're NIH and we're NIH. How have we not had this conversation? And I spoke to, I don't know if Perdita is here, but I spoke to her on Tuesday and I was just like, so we should chat.
And I think that, basically you're almost across the street from each other at
Literally,
NIH.
I also think to address the question, do we need a label change? I think it goes back to what you said. Do we really? Because we're already doing it. And if we're working within the system that we have now, that's just going to take too long. And we have to rethink, I say this all the time, we have to really think outside the box on how we're actually going to do this. So I'm not one who thinks necessarily the label change has to happen. More so, how do we just get the treatments to the patients?
I would agree 100%. I think for me, it comes down to how do we inform correct usage? And on the adult side, it's also how do we convince payers, to allow for drugs? Because some of the drugs we may be talking about are not cheap. And so having the payers at the table would be important, but also I think there's opportunities for NCCN compendium listing, which is often more to allow for usage. And then the other thing that I'll just throw out there for discussion is we don't want to be, we also want to understand what happens in Europe as well, right? Or other countries, because there may be ways to combine data set where they need a label change versus, each country is different. So how do we collectively move forward with this? But I agree. I don't think we have to set the bar to a label change.
Right. But you're right. So we had these conversations with groups in Europe earlier, and for them, because of the way that the payer system is set up there, they absolutely do. Basically, almost everywhere else in the world except the US wants to do that. But I think that we can sort of pull back on it. But then the question has always become, how do we get this information out there? How do we convince physicians to actually that this is better than nothing, and there's definitely some information there for it.
And avoid disinformation, right?
And avoid
Because I
disinformation,
think
exactly.
that is the danger. Because I know we've been knocking our FDA. I have to say I've worked with the FDA for the approval of the MEK inhibitor. It was a wonderful experience. We can feel very confident, despite whatever's going on in the political realm right now, that when the FDA approves something, they have really beautifully gone through the data and, looked at that risk benefit ratio. And so I do, it does provide a level of confidence. And so I do think there is also some worry, because there are, unscrupulous people out there who are taking advantage and saying, oh, again, we saw this during the pandemic, right? Everybody should be taking whatever drug of choice, bleach or whatever.
I'm not going to go there.
I whatever it was. So there's a balance between there, but I do think we need to figure out a way. And again, I think professional organizations, NCCN, Children's Oncology Group are probably the way to go that have that balance.
Any additional thoughts?
And I also think, if you have good evidence, first of all, I think the manufacturer has to seek the label change. So
if there is a manufacturer, that's great.
That's true.
Right.
Yeah, right.
In many of these cases, More than
There
likely,
isn't?
there isn't.
Okay.
And another sort of question that I had is that I know that you mentioned that you had, I think, 7 or 8 or 9 manufacturers came in. I'd like to sort of figure out, how did you do that? Number one, because more than 2 is like,
Right. Right. Yeah. And it's interesting because in one case, one of the manufacturers said to us that they wanted to combine their drug with another company's drug.
Usually they don't say that.
Yeah, I was surprised to hear that too, because they had their own immunotherapy checkpoint inhibitor, but they were interested in data with a different
Different one.
companies. And it was easier to go through us
Yeah.
because they didn't actually have to have an agreement with
with
the other
them.
company. We just went to the other company and said,
This is what we're
hey,
doing.
this is, the other company is telling us they want to do this. Are you interested? And so they did. And we have the agreement with both of them. Now, I don't want to make it sound like it's easy, of course.
No.
But yeah, it does make it, most of the therapies are either monotherapy or combinations that are the same company, but that is possible. And, I think it's definitely opens up the opportunities.
That's inspiring because when you first said that, I was like, I got to ask her about that again.
Yeah. And the study's been open since
Yeah.
2016. So, we had, I think, 7 companies when we launched the study. So we've added a few as we've gone along, but we were able, and that's partly due to, just the number of biomarker driven therapies in oncology. There were a lot of products out there that we were interested in including.
Okay. Any thoughts about, so I think that we're talking about a lot of the ethereal. So if something works, and that's the question, how do you know when it works? Because if you have another group like FDA saying, okay, this is good enough data, but once you don't, then who's going to be the body that's going to be able to sort of say that, other than NCCN or somebody like that?
So I work with the neurofibromatosis community, and I think it's actually in some ways a model community for coming together to define outcome measures for a rare disease, and has both, there's a group called the Response Evaluation in Neurofibromatosis and Schwannomatosis, or RAINS, that was founded by by Scott Plotkin and Brigitte Wiedemann, and they literally brought all the researchers together and and a whole bunch of patient representatives and said, all right, for sleep-related issues in NF, for pulmonary issues in NF, for vision issues in NF, what is the endpoint that's meaningful? And I think that, but that took a ton of work and a ton of organization, and is super, super specific for an individual disease. I don't know how to do it more broadly, but I think bringing in the patient representatives and having them help us define what is meaningful is absolutely crucial. Did I scare Vigil away?
No, she had to leave for her flight. So,
but it's a great point. But the dirty secret is how we make treatment decisions, right? We have, an anecdotal experience from some colleague, or, there's a case report of something, and that's often how we choose what to do in the clinics, right?
Right.
But one of the things that we were talking about earlier is also international consensus and bringing the groups together, the experts with the disease. And it's not, again, just the oncologists. These are multidisciplinary groups, including patients. And you really, if you generate the data and you sift through the data, then you can really decide, what level of evidence do we want to give this? And then what is going to be the expert consensus? And that's something that often happens in rare diseases.
And I agree with you. The thing is, we're talking about rare diseases, so we've got to be more collaborative on an international setting because, it'll take us a decade to get enough patients. So in order for us to be able to do that, we need to partner with as many people as we can.
I think this is one where if you build it, they will come. And I think that's what happened with Taper, watching it from its inception. It's not only that companies saw an opportunity to get maybe their drug used in different indications, but it was also the politically right thing to do, right? You wanted to be at the table. And I think building this in rare diseases, if you can build a platform, more and more people will come. And it's just an opportunity to accelerate things.
Right. And these are all just pilots. So if we can get something to work and stand up, then obviously that will take off and move into other areas. And so that's the plan. I know it's Q2, and I'll see if there are any burning questions from the audience, because if not, I'd like to, unless there's anything that you guys want to say, I'd like to thank the panelists. It was an amazing conversation that we had. Really good conversations in the fact, and having a few people from the audience as well. But thank you so much for coming, and thank you so much for your insight. So before we leave, I think Heather, would you like to say a few words? Oh, okay.
Thank you all so much for coming. This has been such a fantastic discussion. I know this is the oncology session, and people may or may not be interested in this, but I was asked by some colleagues just to remind everyone that the FDA is hosting a patient-focused drug development meeting on long COVID on Tuesday, April 25th. So coming right up from 10 a.m. to 4 p.m. and really encourage all who are here to participate. You don't have to be a patient with long COVID or a provider treating patients with long COVID. But this is going to be, I think, a really important topic in the drug repurposing space, since again, just like with acute COVID, everything that's being used is being used off label. And there's such a dire need for treatments for patients. So please consider attending.
Thanks very much, Heather. And the last thing I'll say is that so this was recorded, as was the previous day. So these last 2 days we'll make available on our website. So we'll send out emails. We'll also send out some contact information so that you don't have to sort of try to find out from LinkedIn, who is that person? What did they say? Whatever. So we'll be able to share that information as well as the slides. So we'll make the slides available. So we'll send that information out to all those that have registered so that you have that material to be able to refer back to. And with that, thank you very much. This is amazing 3 days. Although everybody says you're still standing, I'm like, yeah, actually, it's so energetic. It's energizing to be here with all these people. And I thank you so much, because if you didn't come, it wouldn't have us here. So thank you very much for all of you for attending.